All right, good morning, everybody. Thank you all for the virtual audience, as well as all of our scholars and audience members here in person with us. My name is Dr. Asante Kampalala. I'm a member of the Education and Professional Development team here in the Department of Neurology at Johns Hopkins. And we are really excited to get to present all of our wonderful 2023 Johns Hopkins Neuroscience Scholars Program and NeuroHIV Comorbidities Program Scholars today. They're each going to be giving their presentations one at a time. Please hold all your questions until each speaker is finished with their presentation, and we will go from there. If you have any questions that you'd like to put in the chat, that's also another option. Our first speaker today is going to be Ms. Gabby Chacon. Gabby, if you wouldn't mind putting up your slides, please. Gabby, there she is. All right, Gabby, you can unmute yourself and then please make sure to introduce yourself before you begin. We'll go ahead.
Sure, just 1 second. Okay, perfect. So good morning, everyone. As
All right,
Dr.
Gabby.
Asante, yeah. Can you hear me? Okay, perfect. So good morning, everyone. As Dr. Asante said, my name is Gabriela Chacon. This summer, I had the privilege to work at Dr. Leah's Rubin Brain Health Program under the mentorship of Dr. Asante Kamkwalala. My project will be looking at the impact of stress in women living with HIV, more specifically by focusing on the cortisol awakening response. Now, to give some background information on what the cortisol awakening response, or CAR, is, it's basically a change in cortisol concentrations that happen in the first hour post-waking. Most people show a rise in the cortisol levels upon awakening, which is followed by a progressive reduction over the day. There's evidence that CAR is independent from the cortisol output that we have over the remainder of the day, as there is little association between the concentration of cortisol you would have upon awakening and what you would have throughout the rest of the day. This response is modulated by the interaction of the HPA axis, regional brain activation, and changes in adrenal sensitivity. Previous studies have demonstrated that dysregulation of CAA patterns is associated with immune dysfunction and increased disease susceptibility, potentially leading to an exacerbating HIV-related immune dysfunction in those affected. Normally, our body responds to stress via the hypothalamic pituitary adrenal axis, or HPA axis, in a negative feedback manner. So the way this works is that when we're subjected to stressors, signals are sent to the hypothalamus, which then gets stimulated and releases corticotropic releasing hormone into the anterior pituitary. This signals the release of ACTH down the bloodstream to the adrenal gland, which then prompts the release of cortisol as a response to stress. This leads to a number of changes that helps our body deals with the stressor. Now, when I say that this works in a negative feedback manner, what I mean is that once the cortisol levels reach a really high level, signals are sent back to the hypothalamus and anterior pituitary so that the stress response can be shut down and the cortisol levels go back to the normal state. There's multiple factors that can affect the CAR, including age and gender, time of waking and sleep quality, as well as stress level. And for the purpose of my study, I'll be looking at gender specifically, as well as stress levels. Based on this, the hypothesis of my study is that higher levels of stress in women living with HIV will lead to a more attenuated CAR, as it is shown in a diminished cortisol rise upon awakening. So the data from my study was collected from a much bigger study called the MS-CORE study. This looked at the effects of glucocorticoids in cognition of women with HIV. For this study, there was a total of 4 visits, although the data collected for mine was only focusing on visit 2 and visit 3. And there was a variety of neuropsychological assessment that was performed, one of the most important being the PSS, or the Perceived Stress Scale, in which participants respond a series of questions asking how they feel day to day and to assess their stress levels. In order to be a participant of this study, they had to be a woman who had HIV, aged between 18 and 65 years old. They had to have a PSS score of over 14 and virally suppressed and on CAR. They had to have also some sort of objective cognitive impairment, and they would be excluded from this study if they had used hormone-based contraceptives, if they had schizophrenia or schizoaffective disorders, if they had regular use of steroids, recreational drug use, or closed head injury. To measure the diurnal variations of cortisol of the participants, this was done via saliva collection that was performed at home after visit 2 and visit 3. So at the end of each of these visits, participants were provided with 9 tubes in which they were required to provide saliva samples 15, 30, and 45 minutes post-waking to measure the CAR, as well as 3, 6, 9, and 12 hours post-waking to see the variation throughout the day. And just to give you some data demographics so you guys can see how the group of participants looked like, we had a total of 31 participants with an average age of 54 and an average education of grade 11. Most of the participants were in the age of 51 years old with an education of 12th grade. There was an average CAR for participants of 1.756 and a mean PSS score of 17.87. We wanted to divide participants in 2 groups, being the high stress group and the low stress group. And the high stress, we had 16 participants who had an average CAR of 1.84 and 15 participants in the low stress group who had an average CAR of 1.67. Now, after running the analysis, we found a trend level positive correlation between PSS score and CAR as measured in area under the curve. As you can see right here, in the y-axis, we have the PSS score, in the x-axis, the CAR. While the data looks kind of spread out and not really following any trend, you see that there's a slight positive correlation. And when we look at the R-square value, it was 0.094, meaning that 9% of the CAR was modulated by the PSS score. Moving on, we assess the difference of cortisol levels for both high and low stress groups. So right here, I want to make something clear is this is not following some sort of like increasing trend for the overall CAR. Rather, each time point, it refers to one participant, so the data that we got for each participant. And in the orange line, we have the group of high stress, and the blue line, the group of low stress. As you can see right here, high stress group seems to have a higher average CAR when compared to the low stress groups, which deviates from the initial hypothesis, although it is important to mention that we collected the data from the average CAR, not specifically each time point. And people who suffer from high stress levels throughout the day tend to have higher cortisol levels than normally we would do, which makes sense when we look at the trend that we have. There's going to be a diminish in cortisol levels upon awakening, but this would increase exponentially as the day goes on. Moreover, there was some more analysis performed looking at different confounding variables that perhaps could be modulating the relationship between stress and cortisol levels. We controlled for depression, although the data was non-significant and it seemed to be only relevant in the context of stress and not for the cortisol levels. Although when conducting the analysis for BMI, we found something interesting, as these 2 graphs are basically showing the same, but I would like you to focus on graph A first. When we look at the BMI information and the total CAR, while the data is still pretty spread out, we see like a general trend that as a BMI increases, the CAR also increases for the participants. Now, when we move on to graph B, which divides these time points into the high stress and low stress groups, we see that the red dots referring to the high stress group has a much clearer trend of when the BMI increases, there's an increase in CAR, which is consistent with what has been shown in previous studies. Finally, for my discussion, there is a reduced CARs that is commonly persistent in stressed population, and higher BMI has been previously shown to have a significant inverse association with awakening cortisol. Once again, this is corresponding to what we found and was shown in the previous graph. Our data overall is consistent with previous studies, and the results move in the direction of our hypothesis, although much more research and data analysis will be needed in order to get a more clear idea of what is happening. There were some limitations and there's some ideas for future studies. First of all, and one of the most important points, I believe, is that for future studies, we should assess participants' sleeping schedules, since the MS core study assumed that all participants had a diurnal routine. They didn't take into account whether or not those participants perhaps had a night job and slept throughout the day. And this is really important since it can impact CAR in significant ways. We should also address demographics for a greater understanding of CARs, since the sleep quality and the location where you are can also affect CARs. There's been a bunch of studies that say that all these factors combined can either lead to the increase or reduction of CAR. This plus stress can shed light into a lot of important data that could be used to develop positive treatment for people either with PTSD, depression, or just in general sleep disturbances. Lastly, the analysis should be performed with each individual time point rather than looking at the average total area under the curve. In this way, we would be able to see the trend and how it changes over time throughout the day, rather than just having the general idea. Finally, I would like to thank the Johns Hopkins Neuroscience and NeuroHIV Scholars program, especially Dr. Amanda Brown, Dr. Asante Campolala, and Heather Thomas for the support provided throughout the summer and the opportunity to be here. It's a great honor for me to be part of this. Also to Dr. Leah's Rubin Brain Health Program, especially Isabel Santustis, Chantal Riggs, and Sarah Kanner for making possible this project. Thank you so much.
Thank you so much, Gabby. That was wonderful. You can go ahead and unshare your slides now, please. Does anybody have any questions for Gabby? All right, we're going to move on to our next speaker. All right, next up, we are going to have Amina Dar. Amina? Is she the person? Amina, are you ready? Yes.
Good morning, everyone. My name is Amina Dar, and I'm here to present to you the research I did in automating tortuosity calculations in cerebral blood vasculature. All right, let's try that again. My name is Amina Dar, and I'm here to present my research in automating tortuosity calculations in cerebral blood vasculature. To start with, what is tortuosity? Tortuosity is a measure of how twisted or distorted a path is, and it has medical relevance because it's an indicator of severe hypertension or other illnesses. Currently, estimating tortuosity in fMRI images is a tedious process. Our goal is to provide radiologists with a way of identifying tortuosity in blood vessels completely automatically. In order to achieve that goal, we first want to create a validation set. then develop a bifurcation detection code, compute directional information of the MRI images, find center lines for vessel junctions that we're interested, and then compare the results. In order to create our validation set, we chose 4 vessel junctions to examine: the ICA terminus with ACA and MCA, the MCA bifurcation, the A1 ACOM-A A2 junction, and the basilar PCA convergence. We define a vessel junction as a place where 2 or more vessel branches meet. To the right, we can see an image of a segmentation and slicer. We abstracted centerline coordinates of each junction in a 3D slicer. Here is a circle of Willis, which includes the first 3 vessel junctions that I mentioned before. Can you see my cursor? Yes. This is an example of a centerline of the ICA terminus. We decided to calculate tortuosity using 2 metrics that were common in the literature: the distance metric and the sum of angles metric, as described by Bolid et al. The distance metric is a ratio of the path length of the line segment to the straight line distance between 2 points. In the bottom here, we can see the path length indicated by arrows and the straight line distance given by P0 and Pn-1. You can see that a more curved path would give you a greater ratio and thus a greater tortuosity metric. The second metric we use to calculate tortuosity is called the sum of angles metric. It's simply a measure of all angles on a line segment normalized by path length. After we had completed our validation set, we found these 2 tortuosity metrics in each of the vessel junctions. In order to analyze the morphological information we needed, like tortuosity, we had to divide each junction into the branches which compose them. This is because, as I mentioned, both the tortuosity metric and the sum of angles metric are dependent upon the endpoints that we decided for them. The endpoints of 2 or more branches is ambiguous. Thus, to analyze tortuosity, we averaged the components at each junction. This measurement was then again averaged across 10 subjects to give you the standard deviation and averages you see here, which are consistent with previously published literature. Now that we'd created a validation set, we needed to create a bifurcation detection algorithm. The importance of this might not seem immediate or apparent at first, but in order to avoid any manual endpoint detection, we decided to define vessel branches as the path between 2 bifurcations or a bifurcation and an endpoint. For this reason, we needed a robust bifurcation algorithm. Below, we have a slice of a maximum intensity projection of an MRI image. To the right, we have the results of our bifurcation and endpoint detection code. The red X's are endpoints, while the blue dots are bifurcations. You can see that this code has a 100% success rate in 2D. You may notice that there are a lot of endpoints outside of the main vessel structure, the circle of Willis, but this isn't because of a mistake in the code. This is just because of noise in the original volume, as you can see here. Our next step is to get directional information from the MRI images. To do this, we processed our images using an edited version of Law-et-al's optimally oriented flux, or OOF for short. OOF is a curvilinear structure detector, which is based on image gradient analysis. The image gradient is the directional change in the intensity of an image. In MRI volumes, the highest intensity you can have is 1, and the lowest intensity you can have is 0. Typically, structures like vessels have a much higher intensity than the background. Law-et-al exploited this property in order to give us important information about the directions at each voxel. So what this code actually gives us is 6 values, 3 vectors and 3 scalars per voxel. The largest vector gives us the structure, the direction of the structure at that point, and the 2 smaller ones give us important measurements of centerline prediction. Our next steps are to expand the bifurcation algorithm to 3D, to generate center lines, and to calculate tortuosity metrics for each of the junctions I mentioned earlier and to compare. I'd like to thank my lab and also for Dr. Rathnanathan, Dr. Brown, and Heather, and Dr. Asante for giving me this opportunity to work at the Rail Lab this summer. Thank you. Are there any questions? Why did we? Did you ask why did we choose those junctions in particular? Well, for one reason is that we didn't want to use straight vessels, and there's a lot of variability in how tortuous or winding a lot of cerebral vessels are. You can see that this vessel junction is pretty curved. I don't know if you can see because it's in plane, but it's actually like curving out towards you. We wanted to find junctions that had a high level of tortuosity and not just take something like here, which would be closer to 1, because then you can't really tell if your procedure is working. Thank you.
All right, thank you, Amina. Next up, let me see if I can turn them on. Give me 1 second. All right, next up, we will have another virtual presentation from Labib Mamoon. Labib, when you're ready, go ahead and share your slides, okay?
Okay. Can you see my slides?
Yes.
Okay, great. All right. So good morning. Hello from quarantine. My name is Labib. I'm a rising senior at Colgate University. And so today I'll be talking to you guys about some of my summer research related to NeuroHIV, specifically in the context of the endocannabinoid system and on a cell type called microglia. First, as many of you all, did something happen?
Sorry, 1 second, just trying to get the captions in order. Give me 1 second.
Yeah, sure. Let me know when you're ready.
All right, go ahead, Labib. Sorry about that.
No worries. Okay. So as many of y'all know, HIV in its current state is a very manageable disease, and patients living with HIV are able to be prescribed antiretroviral therapy, or CART for short. And essentially what CART does is suppress the viral load within sort of the peripheral body, but not known to many people is that when that CART is active in the body, it does not do much to sort of act within the brain. And so HIV infection can actually continue to induce inflammation in the brain that can eventually lead to neurologic impairment and the development of disorders such as HAND, or HIV-associated cognitive disorder. And so a big part of NeuroHIV research is to sort of find adjunctive therapies to sort of alleviate some of that inflammation in the brain. And so one way that my laboratory is, one compound that my laboratory is investigating is cannabis. And so patients that actually use cannabis that were infected with HIV have shown to sort of show these decreased levels of CD16 positive monocytes and decreased levels of pro-inflammatory cytokines and chemokines circulating throughout the bloodstream. And so it's an interesting compound to look at due to sort of its anti-inflammatory properties. And so cannabis is comprised of 2 primary components: THC, which is the primary psychoactive component of cannabis, and CBD, which is cannabidiol, and it's known to be the vastly sort of more important anti-inflammatory compound. And it's in many different studies across different models and across different model organisms shown to sort of downregulate levels of pro-inflammatory cytokines and chemokines, namely TNF-alpha and interleukin-1 beta are some examples. And so CBD acts on something called the endocannabinoid system. And the endocannabinoid system is comprised of canonical receptors that you may have heard of, namely CB1 and CB2 are some of those receptors, and they are very well studied. However, not very well studied are extended family receptors are related to sort of different receptors that CBD does interact with, such as the TRPV channels, which are related to capsaicin, and Fa channels, which are related to metabolism. And so all of these receptors are essentially acting and interacting within the extended family endocannabinoid system, but they're lesser studied for their effects in the context of CBD and HIV. And so the cell type specifically that we're looking at is called microglia. You can think of microglia as these sort of resident macrophages in the brain. And they are essentially responsible for the vast majority of inflammatory response that occurs in the CNS. In the context of HIV infection, long-term inflammation and unsustainable inflammation basically occurs where these microglia are releasing many different factors that eventually causes damage to neurons and neurologic impairment. But these microglia also just so happen to express some of the highest amounts of extended family endocannabinoid receptors of any central nervous system cell type. And this is work that we've done in the past where we've essentially cultured a bunch of cells, neurons, astrocytes, pericytes, and we've sort of done qPCR to measure baseline receptor expression. And we found that microglia are really exciting to look at because they express high amounts of these extended family endocannabinoid receptors. So our study essentially is trying to bridge the gap between the role of CBD in immune activation in HIV infected microglia. And so two of the things that we've done to do that is sort of the first of which is sort of to validate a novel macrophage microglia model to sort of simulate the expression of CNS microglia. And so how we're doing this is we're doing something called PBMC isolation. And so we're collecting blood from healthy blood donors, isolating the monocytes within those blood donors and creating cultures of cells that we differentiate into microglia. And so how we're doing this is once we have the blood and we have the monocytes plated in culture, we put things into the media, specifically factors called interleukin 34 and GM-CSF, to differentiate those cells into cell types that we're calling macrophage microglia, which are essentially microglia-like cells. After 14 days, we infect those cells with HIV, and then we do our treatments and assays. And so this is just a brief picture, and I can go into detail afterwards if anybody has questions, but essentially on the left here, you have monocytes, which are very round and circular, eventually becoming microglia as the course of differentiation goes through. And we have data to show that they express differential amounts of microglial specific markers. And so it's successfully essentially showing that microglia, the macrophage microglia that we've cultured, are acting like microglia. And so now that we have a valid model, sort of the second goal that we want to do with this project is sort of to assess the inflammatory response, as well as if there's differential receptor expression given treatments of HIV and CBD. And so we have our differentiated microglia 14 days later after we've done the PBMC isolation. We infect those cells for 24 hours with HIV, and then we're going to do a daily CBD, or we have done daily CBD treatments for a course of 5 days, where we also collect supernatants every day before we lyse the cells. And so with the cell lysates and the supernatants, we run a variety of assays, the first of which being a Legendplex assay, which is a really complicated version of an ELISA, to sort of measure inflammatory response. Also to measure inflammatory response, we're doing Western blot and something called FACS, which is fluorescent activated cell sorting, to sort of measure levels of two substances called NF-kappa B and STAT1. And those are just sort of markers of inflammatory response in the cells. And the last thing that we're doing is qPCR to sort of quantify receptor level changes of extended family endocannabinoid receptor sort of just expression. And so here you can see some pictures. On the left here, you have uninfected microglia with the sort of characteristic elongated morphology. And then on the right here, you have HIV infected microglia, which are just much more bulgy and inflamed. And then once you treat them with CBD, you can potentially kind of see some of that characteristic elongation returning a little bit. However, you can only really speculate, and you need sort of quantitative data to prove that. And so here is just some hot off the presses data that is not, and before I say anything about this, it's not, no statistical analysis has been performed. And so we cannot draw conclusions from this yet. But here is just an example of what CB1 and CB2 look like on some of our cells. As you can see, CB2 is almost not expressed across any cell and any treatment. And CB1 is lowly expressed. And so if you're only looking at the canonical receptors, you only get a small chunk of the portion of the data and a small snapshot of it. Whereas if you're looking at extended family receptors, such as TRPV2 and PPAR alpha, you can see that they're highly expressed across the board, and they're essentially available for the endocannabinoid system and CBD to act upon. And so lastly, we have some FACS data here with inflammatory responses on the graph on the left. I don't know if you can see my mouse, but the HIV treated group has a very high level of STAT1, and so a high level of inflammation, whereas on the purple bar, HIV plus CBD. So when we infect cells with HIV and then do that 5-day CBD treatment, they show a lower level of STAT1 than that of the HIV treated group alone. And so this is only an N=1 sample, and we need more data to be able to prove this, but we can also speculate here. And just it's exciting to see that CBD is reducing levels of inflammation within the cell. And so lastly, why does this all matter? HIV affects 1.2 million people across different races, socioeconomic backgrounds, sexualities, and it's a very complicated disorder. And so it essentially offers patients an ability to take an adjunct therapy that's not necessarily a prescription. You can get CBD treatments from snacks, you can get it from sort of gummies, you can get it from chips even. And so it allows for a treatment that's not necessarily limited to something like Epidiolex or an FDA-approved drug. And it just allows people to sort of manage their overall health better without having to sort of go to a doctor to be able to have that recommendation or prescription prescribed to them. And so all of this data was acquired through many challenges, including literally the P3 flooding, cells being sort of spilled all over the place, and plates of culture. But just a really big shout out to the Williams lab, and especially my mentor, Alicia, for just getting me through the summer and just allowing me to be in an environment where I genuinely was looking forward to come into work every day and happy to see everybody. So yeah. And then thank you to Dr. Brown, Heather, Asante, and Dr. Redenauer for all the work that they've put in over the summer for us to sort of make this program and the symposium happen, despite all the challenges. So yeah, that's it from me. Thank you. Yeah.
All right, well done. We have a question. Hold on. We have a question for Labib from Dr. Brown. You want to come to the microphone?
Sure.
Great job, Labib. I wanted to ask, very interesting with your microglial cultures that have the two morphologies.
Mhm.
Have you tried to distinguish between those two, the more that elongated versus those that are more rounded?
Yeah, so the microglia across the board sort of express a more elongated morphology. The pictures that I showed are essentially, one of the examples was normal microglia that were uninfected, and one of them was HIV infected. And so I think the biggest thing to harp on is that we're not doing a lot of work on sort of fluorescence imaging or sort of the morphological changes, but we are doing work on sort of receptor expression because it's in culture and we're not necessarily looking at cells that are representative fully of microglia. We can sort of speculate that our microglia are representative of microglia and can simulate the CNS microglia that we're trying to replicate, but we call them essentially macrophage microglia because there are sort of some differences that you wouldn't get 100% accurately with sort of like a regular CNS microglia. So, but yeah, I think more work needs to be done with like microscopy. So.
Great, thank you.
Do we have any other questions for Labib? All right, thanks, Labib. Let me pull up our next speaker. All right, next up, we have Alexandra Zandy Wong.
Hello, everyone. My name is Sandy Wong. This is my 4th year with the program. This is my first in-person presentation, so I'm really excited to be here and present. Today, I am talking about work I did this summer. This summer, I contextualized mid-20th century, so that's 1940s to 1970s, efforts to alleviate pediatric hearing healthcare disparities in Baltimore. And this was part of Dr. Amanda Lauer's lab, and I'm lucky to be supported by them. Okay, so some background. In 1945, 4 million children in the United States had hearing loss, and this was from a national study done by the agency at the time, Maternal Health Services, that was the agency of the US government. And historically, childhood hearing loss has been associated with difficulties in developing auditory and vocal communication skills, and also caused academic achievement gaps because they cannot communicate or hear very well. And this happened because the critical age for developing speech and language abilities was between the ages of 2 to 6. So if there were no proper interventions done, such as hearing aids, amplification systems, or speech language pathology, that caused some difficulties in children being able to develop speech and communication abilities. And so as a result, during this time, deaf and hard of hearing children were isolated away from school and community spaces. They were often sent to residential schools, deaf residential schools, which just kept them away from family support and really integrating into community environments. And so going through the path literature, which is a lot of reading in general, I identified the gap that there was a major gap in identifying what the major causes of hearing loss were from the 1940s into 1970s for pediatric and adult patients, although this presentation mainly focuses on pediatric patients. And there was also major gaps in understanding what kind of barriers did children with hearing loss, patients with hearing loss face when they were going into and trying to integrate into school and community spaces. And then during this time from the 1940s to 1970s, there was this married couple named William and Miriam Hardy, who were 2 research audiologists who founded the first hearing and speech clinic in the nation here at Hopkins. It was known as the Hopkins Speech and Hearing Clinic. Although there's not much known about what efforts they did to help children with hearing loss and their families integrate into common community spaces. So I really wanted to learn more about what they're doing or what they did to really help these children integrate into common spaces. And why this research is important, it's not really a quite neuroscience project. It is related to hearing healthcare, which is a big passion of mine, but it's so important to understand the past, understand where we're going now. One of the objectives of the work that I am finishing up right now is that I found the major causes of hearing loss, and we're comparing them to present day causes of hearing loss to see what kind of historical backing do we need to prevent hearing loss going forward in the future. So yeah, it's important. Okay, so some materials and methods. So I spent about a month in June gathering information from the William Miriam Hardy collection at the Chesney Medical Archives in Mount Washington, which is about a 30-minute drive from here. And specifically, the files were retrieved from the Johns Hopkins Institutional Records and the research files. So these were just 2 giant boxes that they gave me, and I spent a month listening to music and reading very old papers that I try not to tear apart. And it went okay, so we'll take it. And the files did not contain personal health information, PHI. And I did not get an IRB for this process because the process just takes a while. So a lot of this data, except one of them, which is cited from a paper, is based off of epidemiological studies and group studies in general. Okay, so what did I learn? First off, the historical causes of hearing loss in the 1940s to 1970s for children with hearing loss were mainly prenatal and right after they're born, natal infections, viral infections. And these infections included measles. Let me get my laser pointer up. Is the laser pointer? Laser pointer. Okay, they include measles, meningitis, and rubella. And rubella and measles were the most studied during this time. And this was a major factor because if the mother was infected with rubella, measles, and meningitis, it was often transferred over to the child, and that caused hearing loss as well. And then some other major causes during the time were apoxia, lack of oxygen during childhood, birth, and birth injuries. And so these 2 graphs, oh, there's Kaylee. Admit. Okay, that's my postdoc, guys. She's awesome. Let's see. So these major 2 graphs, in the x-axis is the causes of hearing loss, and then the y-axis for the graph on the left is the number of people with hearing loss. And this is number of people with hearing loss. This study was contained from the from data collected directly from the Hopkins Hearing and Speech Clinic during the first 2 years of opening. And then the other graph on the right here is also cause of hearing loss on the x-axis and number of people with hearing loss on the right axis. And this was an epidemiological study done by the Cleveland Health Goals Project. And it similarly defined that ear infections and viral infections were a major cause of hearing loss during the time. Okay, what else did I learn? So there was a lot learned about hearing healthcare disparities, but I'm going to go into just one of them right now. So one major issue is that children with hearing loss often slip past being diagnosed with hearing loss because there was issues. Oh, there's my other postdoc. Issues with neonatal hearing screening. So traditionally, children in general are screened within the first day that they're born. And nowadays they use something called auditory brain response, ABRs. But during this time, they use something called electrodermal audiometry, which is like a rudimentary version of ABRs. And so the issue with that is that there were many false positives and false negatives associated with EDR. And this is one example. This is a rubella case. So basically, in this case, the child had rubella, or I think they were infected from their mother. And since the EDR, the electrodermal audiometry, only played at one frequency or one pitch, it basically, okay, I'm not doing a good job explaining this, but in this audiogram, the child has kind of like a bell-shaped curve of hearing. At some frequencies, higher pitch or lower pitch, they can hear better, and at some other frequencies, they can hear a little bit worse. And so the EDR at the time only played at one stimulus, which I think was 1,000 hertz. So it would cause the child to be not diagnosed with hearing loss when they actually had hearing loss. And that was a really bad thing because there was no follow-up afterwards to check if they did have hearing. So besides the issues with trying to diagnose hearing loss in general from EDR audiometry, there was also a lack of audiologists and otologists available to follow up and check the child's hearing, because in rural communities or communities where there just weren't a lot of audiologists in general, there were a lack of resources available. That graph is a little confusing to understand, but I'm happy to explain that. All right, next slide. Okay, and so the last big topic I'm covering is advocacy efforts to address hearing healthcare disparities. So I mentioned the Hardys earlier, and they ended up being really crucial in helping to be the first advocates to help children with hearing loss integrate into school and community settings. They started a hearing and speech clinic here at Hopkins, which served over 4,500 pediatric patients in their first 2 years. And this was directly after World War II, when there was a lot more recognition of hearing loss because of veterans coming home with hearing loss. And then they also published a lot of research papers and worked with congressional staff and briefings to help push forward the notion that kids with hearing loss could integrate into school and community environments as long as they had access to familial support, speech language pathology, and hearing aids. And it is so important that with early exposure to sound and communication, that they could really just be like any other kid. And so they were the first clinicians to really push that forward. And this comic that you see here, the really funny little comic, it was grabbed from a group in California that I looked at. This group of audiologists in California realized that there were rural communities that did not have access to hearing healthcare. So they actually sent out little booklets of how to integrate your kid that was hard of hearing into community settings. And they sent out little comics like this to help the families understand more about hearing loss. Let me go to the next slide. Okay, so some major conclusions. The major causes of hearing loss during the 1940s and 1970s were viral infections, noise exposure, and age. Age is more related to adult patients, but noise exposure and viral infections were a big deal for pediatric patients. And then neonatal hearing screening was not accurate during the 1940s and 1970s, and advocacy efforts really focused on helping pediatric patients with hearing loss get assistive technology and speech language pathology early. And so the next steps of my research are to finish writing up this report and to compare the historical causes of hearing loss to present day causes of hearing loss. And one interesting time point that I want to explore is when the MMR vaccine for rubella, came into play, and see how that affected major causes of hearing loss during that time. Okay, so I want to give a huge thank you to my mentors, Dr. Lauer, Dr. Amanda Brown, Talak, who can't be here right now, but it's always wonderful to have his support, and Heather and Dr. Asante. And this research was supported by this program and also the temporal bone grant out of Dr. Lauer's lab. Thank you so much. Do you guys have any questions? Okay. So the question was, how have the present day causes of hearing loss compared to historical ones I found in 1940s and 70s? So I've just started investigating that data. It's a lot less viral infections. It's more congenital nowadays. Whether that's birth defects from birth, it's similar to that. It's most similar to that. The viral infections have mostly gone away, but I'm still looking at the data. So I'll be able to tell you more later.
I have a question.
Yes, Talak, what's your question?
Yeah, a very good talk. I met Dr. Hardy once in the 1990s. Anyway, the important thing to remember is that we're talking about a paradigm shift from the 1940s, 1950s, and now the profoundly deaf people of the 2020 are actually the hard of hearing people of the 1940s. In other words, the profoundly deaf people in the 1940s never benefited from the hearing technology at that time. Now we see a paradigm shift. So I want you to be aware of that. Okay.
Yep. Thank you, Talak. Yeah, there's definitely a focus on seeing people with disabilities, not limiting them by a disability, but really integrating them into community spaces. So that's really important. Yeah, they weren't the best in the 1940s. Cochlear implants started, Talak can correct me on this, but cochlear implants started coming around in the 1990s. So yeah, it's been a huge shift in just a few decades.
Great, thank you, Zandy. Great talk. Right. Our next speaker this morning is going to be Kevin Uriarte. He's going to be presenting virtually. Let's see if Kevin's logged in. All right, Kevin, good to see you. Go ahead and share your slides. All right, Kevin, you want to go ahead and share your slides, please?
Sorry, 1 second. I'm having a little bit of trouble sharing my slides.
Okay, I have them on my side too, if you'd like me to load them up for you.
Yeah, can you do that for me, please? Thank you so much.
Can you show me where that is? Sorry, I don't see it. So here's the slide first. All right, there you go. Kevin, you should have control of your slides now whenever you're ready.
Thank you so much. So good morning, everyone. This summer, I had the pleasure to work in the Morrison lab, where my main project was the impact of adoptive cell transfer of macrophages engineered to upregulate MCT1 and peripheral nerve regeneration after sciatic nerve injury in mice. So just to start off with some background. So unlike the central nervous system, the peripheral nervous system regenerates. And however, this rate of regeneration, which is around 1 to 3 millimeters per day, may lead to permanent disability for a patient. And this is specifically important for motor nerves because motor nerves have sort of a time limit for healing. The reason for this is because of the structure which has been coined in literature as the motor end plate, which is really a part of the neuromuscular junction where the nerve joins into the muscle. if this so-called motor end plate receives no nerve impulse for around 18 to 24 months, it dies away, and there's no longer any way that this muscle can be activated by the nerve. And it's known that macrophages participate in peripheral nerve regeneration. However, very little is actually known about the intracellular metabolism's role in this function, which is what we are going to be looking at. And then just want to give some quick background on nerve regeneration as a whole. So after nerve injury, the nerve goes into a process which has been coined Wallerian degeneration. And this involves the disintegration of the distal part, the injured nerve, which includes the axon and the myelin sheath. It's known that macrophages and other immune cells play a critical role in actually clearing this debris and cellular remnants at the site of the injury. Then axonal sprouting occurs, where the nerve cell body sends out new axonal sprouts, sort of to attempting to bridge the gap between the injured nerve ends. And then these sprouts are guided by growth factors and other molecules released at the injury site. There's then a Schwann cell response, and these cells are specialized support cells that sort of surround and support nerve fibers in the peripheral nervous system. After nerve injury, these Schwann cells undergo changes that promote this nerve regeneration. And last is the most important part, which is known as target reinnervation, which is where the regenerating axons eventually meet their target tissues, such as muscles or skin. And this reinnervation is critical for restoring sensation and motor function to this affected area. So past papers from our lab have shown that the upregulation of monocarboxyl transporter 1, MCT1, and macrophages in transgenic mice accelerates nerve regeneration, and also conversely that the downregulation of this MCT1 and macrophages decreases nerve regeneration rate. This is also shown in a visual abstract from one of our published papers. You can see that MCT1 overexpression accelerates nerve regeneration, MCT1 ablation leads to impaired metabolism, which impairs nerve regeneration, but also that the introduction of wild type macrophages actually ameliorates this impaired nerve regeneration. So we wanted to take these past results and look at the clinical applicability of MCT1. So we evaluated the effect of adoptive cell transfer of macrophages overexpressing MCT1 on nerve regeneration in mice. So the process of adoptive cell transfer was the main driving point which would have made this process clinically applicable. This process involves taking cells from a patient, in this case, macrophages from mice, and then you culture them, you introduce lipid nanoparticles with upregulated MCT1, and you reinject those back into the mouse, and then you hopefully can see your desired effect. So before we actually started off with the experiment, we had to first confirm this upregulation of MCT1. So for this, we used bone marrow-derived macrophages. We used lipid nanoparticles for the transfection of plasmids, GFP and MCT1 GFP. We injected these cells per animal on day 3 of the sciatic nerve fresh injury. And we did CD11B plus selection, RNA isolation, qPCR, which led to the creation of this figure here, which shows the MCT1 relative expression across 3 groups: the sham group, the lipid nanoparticle GFP group, the lipid nanoparticle MCT1 GFP. And it's clearly shown here that there is an overexpression, a relative overexpression of MCT1 in comparison to the other groups, which allowed us to proceed with the experiment. So just some notes on methodology. So the mice underwent sciatic nerve crush, and then followed by adoptive cell transfer of macrophages with either normal or overexpression of MCT1 3 days post-crush. And the recovery was evaluated over 6 weeks through toast rate index, ladder walk, and electrophysiology. So toast rate index, to sort of describe that, when you grab a mouse, they tend to spread the paws to the best of their ability, so that when the nerve is crushed, they won't be able to spread at all, which gives a score of 0. And when nothing has happened to them, they tend to spread their paws all the way through, which leads to a score of 2. And anything in between is a 1. And then in the figure to the right, you can see the setup for the ladder walk. And we train the mice to walk across a contraption very similar to this. And then we take a ratio of falls to the total number of steps these mice take. And these are just behavioral ways to evaluate nerve regeneration. And I'm not sure if this will work, but I have a video. If you can play that. It's okay, it wasn't essential. Thank you. Essentially, this video, I was just trying to picture the sciatic nerve crush injury. So yeah, so the sciatic nerve crush injury, where essentially you would make an incision into the mice, you find the sciatic nerve, you separate it from the muscle fascia, and then you crush it with a pair of blunt forceps for around 20 seconds. And this obviously is one of the main parts of the experiment that we are doing to evaluate this nerve recovery after this injury. And moving on to the results, so a total of 29 mice were used in this experiment. 10 mice were a sham group, which were injected with PBS control. 10 mice were injected with macrophages, induced to express GFP, and 9 mice were induced with macrophages to express MCT1 and GFP. So here you can see our 3 figures. The first graph shows percent lateral falls versus days post-crush. Figure 2 shows the graph. It's a graph showing toe spread index versus days post-crush. And then figure 3 is a toe spread index, specifically on day 12, where we saw a difference between the lipid nanoparticle GFP macrophages and the MCT1 overregulated macrophages. However, as you can see in figure 1, there isn't much change shown between any of the groups, and similarly also in figure 2. I don't think I have control anymore. Thank you. So just some conclusions. So further experiments are obviously needed to explore the effect of this adaptive cell transfer of macrophages with upregulated MCT1 on peripheral nerve system regeneration. And based on our preliminary data, there is no significant improved recovery in nerve regeneration. However, additional data, which includes electrophysiology and histology, are still being completed and may change final conclusions. And then just acknowledgements. Everyone in the Morrison lab, Dr. Morrison, Atul, Sagar, Fong, they made this experience really great and have made it really easy to learn, which has been amazing. And obviously everyone in the program here, Heather, Dr. Brown, and Asante. And that's it.
Thank you, Kevin. Does anybody have any questions? Yeah. Dr. Brown's question was, do you have any information about whether the day of the delivery of the crush injury needs to be changed, whether it should, the macrophages, excuse me, whether it should be earlier or later?
Well, I spoke with my postdoc about this, simply because we saw that that change at day 12. And the reason we actually do it after 3 days after sciatic nerve crush injury is because we want to get a baseline for all of our different techniques, both before the nerve crush injury, but also after, so we can have a reliable way to track nerve regeneration over time. And if we're looking to get the day 12 spike, which I think I heard Dr. Brown mention, there isn't much that can be derived from that. When speaking with my postdoc, we spoke about maybe taking earlier time points to see if we can further quantify that spike and see if we can see exactly what day it occurred instead of having sort of a 6-day gap to where it could have occurred. So a different way to evaluate that would be to take multiple points, so either every 2 days or every day leading up to that point. But in terms of when these macrophages are injected, there isn't, I don't think we needed to change it at all. There is, I guess, the possibility of injecting macrophages upregulate MCT1 later on. However, in literature, it's shown that around day 14 after injury, macrophages are at their peak. So I don't know if that will really have any effect. It might even have a negative effect on the mice. Did that answer your question?
All right, great job, Kevin. Okay, next up, we have a break. It is 9:39 now. We'll be back right at 9:50 to continue our presentation. See you all then. Dr. Brown, are you ready?
That's a good one. But let's wait. Big pause. Everybody in. Pause.
All right, welcome back, everybody. Thank you so much for sticking with us. We're going to start up with the next block of presentations, and up next is Tori Fergus.
Hi everyone, today I'm going to discuss how plasma IP10 levels correlate with brain TSPO binding in virally suppressed people with HIV. Okay. Despite successful antiretroviral therapy, HIV-associated neurocognitive impairment remains prevalent. One translocator protein 18-KDA, otherwise known as TSPO, is a biomarker of the neuroimmune response of immune cells such as microglia and reactive astrocytes. We previously reported high TSPO in brains of virally suppressed people with HIV associated with lower performance in some cognitive domains. Therefore, we aim to determine if pro-inflammatory cytokines or chemokines in the plasma correlate with TSPO binding in the brain of virally suppressed people with HIV. Monocytes are a form of white blood cells that inhabit the blood and tissues with the purpose of destroying pathogens and eliminating infected cells. And astrocytes are a subset of glial cells that inhabit the central nervous system and have a responsibility of regulating the blood-brain barrier. And by looking at astrocytes, we are able to better understand how pathogenic factors impact and harm the blood-brain barrier and consequently lead to neurodegenerative side effects of HIV, such as neuroinflammation. Objective: we aim to determine if pro-inflammatory cytokines or chemokines in the plasma correlate with TSPO binding in the brain of virally suppressed people with HIV. Pro-inflammatory chemokines are a form of cytokines, which are proteins that are secreted by human immune cells that attract white blood cells towards the sites of infection and regulate the growth of these white blood cells. And we focus on the plasma because it is a soluble fraction of the blood. CSF, or cerebral spinal fluid, is another soluble fraction of blood, but CSF poses a higher risk to acquire from individuals, which is why we didn't use it. And hypothesis: we hypothesize that chemokines associated with the recruitment of peripheral immune cells may be associated with high brain TSPO binding in virally suppressed people with HIV. And these peripheral immune cells consist of lymphocytes and monocytes that migrate into the central nervous system and are involved in inflammatory lesion development. Materials and methods: 25 virally suppressed people with HIV and 18 demographically similar HIV uninfected donors were enrolled. The goal was to look at a broad panel of pro-inflammatory analytes. This includes chemokines and cytokines, some that have been known to have antiviral effects and others that have been expressed chronically. First, each participant completed PET scans with arterial blood collection to assess binding potential in 9 brain regions. The study aimed to explore the relationship between higher frontal brain binding potential and lower cognitive/behavioral performance in HIV patients on antiretroviral therapy. And PET works as a fluorescent tracer that utilizes TSPO as a marker of neuroinflammation, which shows a positive relationship with activated microglia and astrocytes, responding to neuronal damage in the central nervous system. TSPO is a receptor expressed on immune cells, and the immune cell binds to a portion of the receptor and light is emitted. And the greater the binding, the more light is emitted. And then plasma samples were analyzed using MSD technology or mesoscale discovery. And the immunoassay used biotinylated capture antibodies, electrochemoluminescent labels, and muplex linkers. And the MSD instrument measured emitted light intensity, providing qualitative data for each analyte in the sample. And the analytes assessed were interferon beta, gamma, interferon 1, IP10, IL-6, IL-15, IL-18, and TNF. Brain TSPO binding is elevated in virally suppressed people with HIV and differs with genotype. We found that TSPO binding expression was statistically elevated for patients with virally suppressed HIV as opposed to those that were healthy donors, and that there are two different types of genotypes of TSPO binding. We have the CC genotype and the CT genotype. And the difference of a genotype is caused by a nucleotide polymorphism, otherwise known as SNP. And this is a form of genetic variation that represents a difference in a single DNA nucleotide, in this case, either cytosine or thymine. And people with CC binding have higher potential or higher binding potential for TSPO, while people with CT have lower binding potential. Brain TSPO binding is correlated with plasma IP10 levels. Our data suggests that there is a relationship between TSPO binding that correlates with plasma IP10 levels, and this means that higher levels of TSPO binding expression indicated higher levels of plasma IP10, and lower levels of TSPO binding expression indicated lower levels of IP10 in the plasma. And R value of 0.4 suggests that there is a moderate correlation between the TSPO binding and IP10 levels, and a p-value of 0.008 suggests that there is a statistical significance behind this correlation as opposed to a random occurrence. Plasma IP10 levels are higher in virally suppressed people with HIV. We obtained a p-value of 0.01, suggesting statistical significance between the IP10 levels of patients with virally suppressed HIV as opposed to those that are healthy donors. And our results indicate that being HIV positive predisposes you for having higher IP10, but sex does not predispose an individual for having higher levels of IP10 in plasma. And then IP10 levels in plasma correlate with brain TSPO binding, but only in individuals with virally suppressed HIV. We looked at IP10 levels by stratifying the serostatus and evaluating correlation between IP10 in plasma and brain TSPO binding between patients with virally suppressed HIV and those that were healthy donors. And we found that IP10 levels in patients with virally suppressed HIV had a moderate to strong correlation with an R value of 0.45 and statistical significance with a p-value of 0.02. However, we found that there was a weak correlation between IP10 levels and TSPO binding for healthy donors, with a weak R value of 0.29 and a non-statistically significant p-value of 0.23. Overall, our results suggest that plasma IP10 positively correlates with TSPO in brains of virally suppressed people with HIV. And IP10, also known as CXCL10, is a chemokine that is associated with the trafficking of immune cells to the central nervous system. Further work is needed to determine if IP10 drives neuroinflammation or is a result of the inflammation. Limitation of the study include that we analyzed only a small set of cytokines and chemokines, despite being a large PET study, and there was a small interim of cytokine analysis that can be expanded on in the future. Next steps from this research regard evaluating if IP10 influences the TSPO binding, or does the TSPO binding influence levels of IP10? And determining the relationship of IP10 and brain TSPO binding will aid in the development of novel therapeutics that target central nervous system inflammation. Additionally, we evaluated 17 more analytes. We only found a significant correlation between brain TSPO binding and MCSF. MCSF is a growth factor that regulates the proliferation, differentiation, and function of monocytes. And we also found a correlation between levels of MCSF and IP10 levels. Future directions include looking more deeply into the relationship between these two analytes. Acknowledgements. I would like to thank Dr. Wienhuis, the Wienhuis Clements Lab, Dr. Brown, Dr. Asante, Ms. Heather, the faculty and staff of the Johns Hopkins Pet Center, participants of the IMPACT study, as well as the IMPACT Grant. Thank you. Questions? No, there was no, we found that there was no statistical significance or that about HIV and TSPO binding for depending on like sex. Yeah.
All right, excellent job, Corey. Thank you so much. Next up, we have Diego Rodriguez.
Hey everyone, my name is Diego, and I'll be presenting my presentation, Developing Molecular Imaging Biomarkers for Radiotherapy-Induced Brain Injury. So, therapies for brain tumors frequently have serious adverse effects that impair patients' quality of life after treatment. In particular, radiotherapy shows a strong correlation with permanent cognitive decline and neurological impairments. Radiation-induced brain injury, is also known as this, can lead to anatomical and functional deficits with symptoms reported as acute, early delayed, and late delayed. Therefore, developing non-invasive imaging biomarkers are very important for early therapeutic intervention and to monitor responses to therapy to avoid future cognitive impairments in patients. So a little bit of what has been done. We've developed a non-invasive MRI biomarker and tested it on mice, but the MRI corresponded with poor outcomes of survival. As you can see with the red arrow, it shows how hemorrhage increases over time, months post radiation. And figure B shows hypointensivity of these mice post radiation. So two hypotheses are taken into account, the parenchymal hypothesis and the vascular hypothesis. The parenchymal hypothesis argues that oligodendrocytes, which are cells needed for the myelin sheath on the neurons, are affected during radiotherapy and therefore depleted, which leads to demyelination and then white matter loss. Then the vascular hypothesis argues that vascular damage occurs, which not only leads to oligodendrocytes being depleted, but also to ischemia and then white matter loss. So basically, white matter loss will happen regardless. But I'm only focusing more on the parenchymal hypothesis because I'm focusing more on the demyelination part of the project. So for this studies, we used six-week-old mice, which were then induced with irradiation in a custom-built small animal radiation research product. And all the mice used for histology and immunohistochemistry were perfused with a 1% heparinized phosphate preferred saline solution, then a 4% paraformaldehyde solution. The brains were then paraffin embedded and sectioned into 4 micron slices. The tissue sections were then stained with Pearl's Persian blue, PBB, HNE, which is hematoxylin and eosine, HNE, and also with others like oligodendrocytes, which is the green, OLIG2, and myelin basement protein, which is the red. They were the Pearl's Persian blue and the HNE were stained with, were lighted with light microscopy, and the others were imaged with fluorescence microscopy. And we also imaged all of them together at the bottom. So the Pearl's Persian blue will focus on how much hemorrhage or iron is built in the brain post-radiation. After 4 months, the irradiated hemisphere of the brain shows like blue stains. After 6 months, it shows more blue stains, and after 12 months, it shows even more blue stains, indicating that the non-invasive radiotherapy is actually damaging the brain. Then the H&E shows lack of cellularity. 4 months post-radiation, you can see that the irradiated hemisphere has more white spots than the non-irradiated hemisphere, along with the 9 months and the 12 months. And the myelin basement protein and the oligodendrocytes were basically show where the myelin in the brain is. However, we are expected to not see these proteins in the irradiated hemispheres post-radiation, since we hypothesized that demyelination occurs in the irradiated hemisphere. But as you can see here, the irradiated hemisphere is showing the MVP and the OLIG2 at the same time. Here is the quantification of the pearl-Persian blue, showing an increase of hemorrhage post-radiation, and the quantification of H&E showing also increase of lack of cellularity post-radiation. The good thing is that the quantification of the pearl-Persian blue also correlates with the hypointensivity of the mice who were first irradiated with the non-invasive biomarkers, showing that there is damage in the brain. So overall, we successfully showed a correlation between non-invasive MRI biomarkers of late injury that were indicative of poor survival outcomes with histological biomarkers indicative of loss of cellularity of cell, which is H&E, and vascular damage or hemorrhage, which is pearl-Persian blue. We also show changes in the expression of both myelin basement protein and oligodendrocytes in the irradiated brain hemisphere compared to the non-irradiated hemisphere. But these studies are still ongoing. So as I said before, the MVP was not supposed to be showing on the irradiated hemisphere of the brain. So this is something that is still ongoing and we're not sure about what is actually going on. And so what's next? For future studies, it will involve better characterizing the changes in MVP and oligodendrocytes expression detected after irradiation. So this model will be used to validate the efficacy of treatments of RIBI. And also MRI and PET scans will be used to further validate the trends we're already seeing. And once we have this model more established, it can be used as a benchmark to compare therapies. And none of this could be done without the help of Dr. Ethel Gen, which is my mentor, Maya, who I've been working with, and of course, Dr. Brown, Ms. Heather Thomas, and Dr. Asante, who gave me the opportunity to be here this summer. Okay, so this yeah. Oh, she's asking which image represents what. So the pink ones are Persian blue, shows hemorrhage of the brain. The purple ones are H&E, show lack of cellularity of the brain. The blue one is just DNA marker, which is just used for correlation of the a control for the brain. The green one is oligodendrocytes, shows where oligodendrocyte cells are in the brain. And the red one is myelin basement protein, which shows where the myelin is in the brain. And the last one is just all of them imaged together. Yeah, so. Yeah, so the brain was irradiated on only one hemisphere, and MVP, which is the red, is supposed to show where myelin is. And since we theorized that demyelination is occurring, we're not expecting to see any of the red on the irradiated hemisphere of the brain. But this is not the case in here. So this is a study that is still ongoing. Oh. Yeah. Yeah, there's from 2 weeks to 12 months post-irradiation. I think that maybe the myelin basement protein that it's being used is the one that's actually trying to replace the myelin, which is why it's showing, but I'm not really that sure.
All right. Next up, we have Brandon Villanueva Sanchez. Come on up.
Morning, everyone. My name is Brandon Villanueva Sanchez. Hi again. My name is Brandon Villanueva Sanchez, and this summer I had the opportunity to work in Dr. Gregory Carr's in vivo pharmacology unit over at the Lieber Institute for Brain Development under the direct supervision of staff scientist Dr. Karen Sita. And my presentation is titled Assessing Real-Time Dopamine Release in the Prefrontal Cortex in Mice During a Food Acquisition Assay. And the goal of this project was to validate the use of an already validated tool, fiber photometry, at our institution by attempting to replicate and expand on the results from an already established study. And the study we chose to replicate is titled Tocopone Enhances Food-Evoked Dopamine Efflux and Executive Memory Processes Mediated by the rat prefrontal cortex. But we were focusing on recording dopamine levels in the prefrontal cortex of mice throughout a food anticipation assay, particularly in the final 2 stages of the assay itself. And the assay consists of 4 distinct stages. The first being a baseline where a mouse is placed into the chamber and allows the instrument that we're using, fiber photometry, to read a baseline level of dopamine activity. Then it is followed by an acclimation stage where at the beginning of the stage, mice are given an intraperitoneal injection of either amphetamine, tocopone, or saline, and then allowed to acclimate for an hour to an hour and a quarter. And then the third stage, our first stage of interest, the anticipation stage, the mice are given visual and olfactory cues of food, meaning they can see and smell food, but aren't given access to the food. And this stage lasts for 10 minutes, followed by the consumption phase where the restricted access is taken away and they are given unrestricted access to the food for the following hour. And the way we went about doing this was we used 12-week-old male mice and who underwent viral transfusion and stereotactic surgery to implant a 400 micrometer fiber optic cannula into their prefrontal cortex. And following recovery, they were food restricted to 85 to 90% of their ad libitum or free eating body weight. And for the food anticipation assay itself, they underwent the assay 1 time per week, and the intraperitoneal injection was either amphetamine at a dose of 3 milligrams per kilogram, tocopone at 30 milligrams per kilogram, or vehicle, which had a volume of 10 times their mass in grams. And tocopone is what's known as a COMT inhibitor. And COMT is the main metabolizer of dopamine in the prefrontal cortex. So we were using this as a method of visualizing various signals of dopamine. And data was analyzed using the guided photometry analysis in Python, otherwise known as Guppy, for data processing, and then MATLAB was used for data plotting. And the results, so I've mentioned fiber photometry, but I haven't actually outlined what it is. And fiber photometry is a tool that's based in optogenetics, and it is a tool that measures changes in fluorescence signals which correspond to cellular dynamics. And one great benefit of fiber photometry is it allows us to measure real-time cellular dynamics of single or multiple neural circuits. And for our case, we use genetically encoded sensors, particularly dopamine sensors, to measure changes in extracellular dopamine concentrations. Here I've included a little figure of how fiber photometry more or less works, but I don't necessarily have time to get into the exact details, but I'd be more than happy to answer any questions afterwards. And these were the results that we were wanting to replicate or attempt to replicate. And this, what we see here is throughout the food anticipation assay, we see an increase of dopamine at the anticipation and the consumption phase. The anticipation phase is outlined by the 2 vertical bars with the consumption phase following right after the second vertical bar. However, what we see here is we see for the tocopone treated group, a near linear increase of dopamine, and again, same thing for saline. And another thing we see is the animals treated with tocopone display a larger initial dopamine measurement with a prolonged duration. However, this study used a technique known as macrodialysis, which has been validated to record neurotransmitter or neurochemical levels. However, it lacks temporal resolution. So they were only able to record measurements every 10 minutes. And for our results, we found consistent results. We found that topical-treated animals display larger initial dopamine measurement levels along with prolonged duration. However, because we were able to see real-time changes in the dopamine concentrations, we saw an individual increase at the start of the anticipation and the consumption phase, something that is outlined here in these two graphs. So the graph on the right displays the data for the entire 10-minute anticipation phase, and the graph on the left displays the data for the consumption phase for the first 2 minutes of the consumption phase. And each graph has its own individual peak, something that wasn't seen in LAPISH. And it's important for me to say that this data was recorded from trials where the measurement tool was turned on and off at intervals to avoid an effect called photobleaching or degradation of the sensor that we were using. But afterwards, we asked, what if we could record the entire food anticipation assay? And so we did exactly that. And this graph comes from a mouse that was treated with amphetamine. Amphetamine in this case served as a positive control, meaning because of amphetamine's mechanism of action, it increases dopamine everywhere it goes. So we expect to see an increase of dopamine after the intraperitoneal injection of amphetamine, which is exactly what we see in the acclimation stage, with peak dopamine levels reaching about halfway throughout that acclimation stage. And even though dopamine levels began to fall after that, at the beginning of the anticipation and the consumption phase, we see the two individual spikes again, something not observed in LAPISH at all. And to conclude, we were successfully able to use fiber photometry to replicate and expand, to replicate and record continuous dopamine transmission levels throughout the entire food anticipation assay. And what we saw was two separate individual increases in extracellular dopamine observed at the start of the anticipation and at the consumption phase, something once again not seen in LAPISH et al. In 2009. And successful replication of this study paired with continuous recording of dopamine transmission levels validates fiber photometry as a molecular tool to be used at the Lieber Institute for Brain Development. And the Lieber Institute focuses heavily on schizophrenia, and one of the areas it focuses on is cognitive deficits. And cognitive deficits in individuals with schizophrenia are thought to be related to dopamine in the prefrontal cortex. So the validation of this tool opens research to observe dopamine transmission levels of various compounds that are made in hopes of rescuing the cognitive deficits of individuals with schizophrenia. And not only that, the use of fiber photometry allows for an increased understanding of the pharmacodynamics of potential therapeutics, hoping to increase the effects to better the deficits of schizophrenia. You know what I mean. So with that said, I want to acknowledge Dr. Gregory Carr, who luckily was able to make it here today, for having me this summer, the past 3 summers, and everyone in his lab for making me feel very welcomed and helping me. Chemistry department, Ingrid, who helped create a few solutions for me, and the directors of the Neuroscience and the NeuroPhytes program, Dr. Maynard Brown, Asante, Tilak, and Heather, for their support and everything they have done for us the past several years, and the professional development team, Dr. Claire Carter and Kathy Hansen, for preparing us for to enter into the real world. So with that, thank you. Any questions? Yes. Yeah, so to my knowledge, okay, so she asked why we only used male mice, and she was wondering if there's a sex difference. I can't speak to a known sex difference that comes off the top of my head. However, we did only use male mice because the study that we decided to replicate, LAPISH, also only used male mice. So, yes, Shaheen?
What is special?
Yeah, so going back to the results from the study, so the food anticipation assay offered a unique paradigm in that the two stages, the consumption, the anticipation, and the consumption phase were fairly close together. So even though other studies have used microdialysis, they weren't able to actually record rapid changes in extracellular dopamine concentrations. So because that was something not seen, fiber photometry offered us the ability to see those rapid changes in dopamine because of the various stages of the assay itself.
Next up, we're going to have Ana Arzeno.
Good morning. My name is Ana Arzeno, and this summer I've had the privilege to work alongside Dr. Eileen Scully at the Department of Infectious Diseases. This past few weeks, I have been studying the effect of estradiol on estrogen receptor splicing and function in human monocytes. Despite more than half of the 38 million people living with HIV worldwide being women, the influence of biological tests has not received significant attention in HIV cure research, evidenced by the underrepresentation of women in clinical studies. Combination antiretroviral therapy effectively reduces HIV replication in cellular reservoirs, in monocytes, T cells, and macrophages. However, it fails to eradicate the virus due to latent cellular reservoirs in T cells. These reservoirs contain dormant HIV genomes that can reactivate years after infection, making their depletion a crucial goal in HIV cure efforts. Understanding the reservoir's nature and the role of biological sex is essential for successful HIV eradication protocols. Notably, studies have shown that estrogen represses latency reversal, and blocking estrogen receptor alpha enhances reactivation. In addition, unbiased small hairpin RNA screening has also identified the estrogen receptor as a potent inhibitor of HIV transcription in latency models and primary cells. Such findings offer potential insights for a kick and kill strategy to eliminate latently infected cells. In this figure here, I try to demonstrate what would be the kick and kill strategy. The kick and kill method proposes to reverse latently infected cells such that they can express HIV. Reactivated cells can then be targeted and killed by the immune system or by antiretroviral therapy. Estrogen receptor alpha, or ER alpha, is one of two main types of estrogen receptors. It is a ligand-dependent transcription factor that is activated by the binding of the sex hormone estrogen. In humans, ER alpha is encoded by the gene ESR1. Importantly, the ESR1 protein consists of several functional distinct domains formed by different exons. Alternative splicing of ESR1 can lead to variants of ER alpha with variant functions. Thus, our lab has been characterizing the variants of ER alpha present in different immune cells, as well as the relationship between estradiol, a form of estrogen, and displacing variants. My work has been focused specifically on monocytes. Our study focuses on main three objectives. The first one is to determine the isoforms of ESR1 present in monocytes. The second one is to identify the molecular weight of ER alpha found in monocytes and its cellular localization. The third one is to determine the impact of estradiol on monocyte gene expression in vitro. To assess the first objective, we first reverse transcribe total RNA from monocyte samples of 3 males and 2 female samples. The samples have been previously stimulated with media, estradiol, LPS, and co-exposure of estradiol and LPS. LPS was chosen as a condition as it is widely recognized as a potent activator of monocytes. After reverse transcription, we employed 2 rounds of nested PCR to generate ER alpha products spanning the coding domain. In the third nested PCR, we used primers from a study by Perlman et al., in which we targeted specific ESR1 splice variants. The name of the variant signifies the specific exon it is missing, such as the variant Delta 2 is lacking exon 2 of ESR1. By sequencing PCR products, we detected numerous variants of ESR1 present in monocyte samples. Here, I showed the results by condition of one of our samples. Although there are multiple bands present in each well, the red box identifies that the expected base pair length was present. Interestingly, the presence of ESR1 variants seems to be partially differently expressed by condition, but with no clear pattern. In the table to the left, it is summarized all our findings from the nested PCR procedure. Consistent with previous studies done on PBMCs and T cells, our monocyte samples express Delta 2 and Delta 7 in all of the conditions. Additionally, individual variation in the isoforms Delta 4, Delta 5, and Delta 6 were present. Lastly, Delta 5 expression may be partially affected by condition. In the table to the right are detailed the expected base pair length of the full-length ESR1, as well as the variants we tested. To assess the second objective, we isolated monocytes from PBMCs and stimulated them in estradiol and media conditions. In addition, we also cultured MCS7 breast cancer cells in the same conditions to function as a control, given their very concentrated ER alpha concentration. Then we separated the nuclei and cytoplasm of the simulated monocytes and MCS7s. Following, we performed an SCS page and Western blot analysis, proing for ER alpha, such that we could identify the presence of the protein on each sample. The latter is especially important as we needed to see where it could be cellularly localized. Our preliminary results show that ER alpha is localized in the cytoplasm and nucleus of our sample of male monocytes. Previous results from our lab have shown that ER alpha is only in the cytoplasm of male T cells. Most interestingly, the molecular weight of ER alpha in monocytes and T cells does not correspond to the weight of the full-length protein, which is around 66 kilodaltons. Based on protein band size and PCR products, different ESR1 variants could be responsible for the ER alpha in our samples. To assess our third and last objective, we performed RNA-seq analysis in 3 male and 3 female samples that were stimulated with and without estradiol. This technique is useful for identifying differences in gene expression mediated by conditions. Shown in these 2 graphs are previous results in which estradiol stimulation had minimal effects on gene transcription in T cells. However, data from RNA-seq done in our monocyte samples show that estradiol stimulation impacts gene transcription in female monocytes, but not in male monocytes. Given our previous results in which male monocytes, in which ER alpha is localized in our male monocyte samples, we're surprised about the minimal effects estradiol had on gene transcription. However, our experiments were all done in small sample sizes, and our results could be a reflection of this limitation. Likewise, another interesting explanation for the discrepancy between results is that the variance of ER alpha found does not function as a transcription factor. More research is needed to have more clear results. So going back to our study's objectives, we have found that splice variants of ESR1 are present in human monocytes, and one variant appears to be differently expressed by condition. We also found that ER alpha localization is different in male monocytes, male T cells, and MCS7 cells. And lastly, the estradiol exposure in vitro impacts gene expression in female monocytes. Some future directions for the studies are to confirm protein sequence of the variants of ER alpha present in our samples via mass spectrometry, and to identify the pathways affected by estradiol stimulation in monocytes via RNA-seq pathway analysis. I would like to acknowledge everyone in this program, in particular the directors, Dr. Amanda Brown, Ms. Heather Thomas, and Dr. Asante. And I would also like to acknowledge my mentor, Dr. Eileen Scully, who couldn't be here today, but has helped me all along this summer. And the PhD students that worked with me all the summer, Erin Mahutzik and Anna Ward. Thank you.
Next up is going to be Alondra Betancourt. Looks like yours is not in full. Bear with us just a moment, everyone. Just loading up her slides.
Good morning, everyone. This summer, I had the privilege of working with Dr. Severance at the Stanley Division of Developmental Neurovirology, and my project is titled Tracking Children's Mental Health in a Post-COVID-19 Urban Clinic, How Behavioral Tools Single Out the Critical Role of GI Wellness. Let's stop share. As a first response to invading agents, the immune system sends inflammatory cells and cytokines illustrated in this diagram. Although we typically see inflammation occur in the form of heat, redness, swelling, pain, and loss of function, gastrointestinal inflammation may also occur as a response to pathogens, damaged cells, and irritants. With increasing degrees and duration of systemic inflammation, the blood-brain barrier also becomes more permeable. We also produce antibodies specific to certain antigens, and they are naturally produced by plasma cells. The antibodies produced by a body tells us a history of a body's previous exposure to certain pathogens. Immune cells and enteric glial cells sense microbiome-associated molecules produced at the gut level. In response to these stimuli generated by the microbiome population, neuromodulator molecules targeting the enteric nerves can be secreted locally. Once the enteric nerves are stimulated, they can in turn affect the brain and spinal cord through vagal transmission. These interactions help explain the mechanisms underlying the microbiome gut-brain axis in different pathologies, including neuropsychiatric disorders. And in this figure, I illustrate how the gut interacts with the brain through the vagus nerve. This project focuses on COVID-19, which is a variety of coronaviruses, which is a group of related RNA viruses that cause diseases in mammals and birds. COVID-19 remains unexplored by much of scientific literature, despite its impressive impact on society in the past 3 years. Thus, our goal is to evaluate if an unstable pro-inflammatory gut-brain axis contributes to childhood behaviors and future mental health in a post-COVID-19 setting. The methods of this study begins with designing a series of neurological and gastrointestinal biomarkers to preliminary screen a set of baseline serum samples from children. And these children come from Baltimore, and they're known to have low socioeconomic backgrounds, which is really important for us to understand how social determinants of health can help us understand how children were disproportionately affected by the pandemic. We screened these samples using the ELISA method, and the ELISAs that I did this summer include fatty acid binding protein 2, soluble CD14, LPS binding protein, calprotectin, casein, Candida albicans, and anti-Saccharomyces cerevisiae IgA or IgG. We screen mental health of a child by the child behavioral checklist, and we analyze all of our data via STATA. The last part of the study will be collecting gut microbiome specimens from the children. The ELISA is an immunological assay used to measure antibodies, antigens, proteins, and glycoproteins. I carry out these assays in special absorbent 96-well plates, allowing multiple samples to be measured in a single experiment. The steps of the ELISA are outlined here, and typically an ELISA just has an antigen or an antibody on a plate, and then the antigen or antibody in the sample is attached to that once you put it in. And after a few washing steps, you can measure the optical density of a plate, and it looks like this once I finish an experiment. And the optical density correlates with the concentration of antibodies or antigens in a certain sample, and I read it on a microplate reader. I also mentioned how we use a child behavioral checklist to assess mental health, and the child behavioral checklist is a component of the Achenbach system of empirically based assessment that is used to detect behavioral and emotional problems in children and adolescents. We administer these tests to 6 to 18-year-olds. However, for children from 6 to 11 years old, they're completed by parents. The test has around 113 questions scored on a 3-point scale, and the categories it assesses are aggression, attention, conduct, defiance, depression, rules, social, and thought. Dr. Severance and I employed preliminary statistics to analyze our data. The one-tailed p-value is used to measure the strength of the evidence against our null hypothesis, and a small p-value less than the pre-specified alpha level of 0.05 indicates strong evidence against the null. Thank you. Our first t-test between the GI biomarkers and COVID-19 positivity demonstrated a significant difference between the concentration of Candida albicans between COVID-19 positive and negative patients. Candida albicans is a pathogenic yeast that is a common member of human gut flora. Previous studies have actually observed similar patterns for fungal infections. I highlight here how there is a p-value less than 0.05 for Candida albicans only, while all of them are much greater than 0.05. And we also use assays for antibodies to the nucleocapsid protein of SARS-CoV-2 and the spike protein to detect differences in Candida albicans levels between COVID-19 positive and COVID-19 negative children. And both assays showed that, that Candida albicans is greater in concentration in COVID positive patients. And the nucleocapsid protein is on the inside of the coronavirus, whereas the spike protein is located on the outer membrane. So this was really just 2 ways to reinforce the results that we found. And this is how the output from STATA appears. And here we see potential relationships between COVID-19 positivity and Candida albicans with whole casein and age, COVID-19 positivity with whole casein and calprotectin, COVID-19 positivity with soluble CD14 and Candida albicans. And I highlight here everything that I just mentioned, and you can see here that there is always a p-value less than 0.05 typically, and the relationships between each of them are highlighted here, and you analyze, this is the main analyzation tool that you're using. However, we did not put seronegative individuals in this because they did not have significant regression coefficients, meaning that their p-values were greater than 0.05. Also, in the negative groups, we see that negative correlations coefficients insinuate beneficial relationship with Candida albicans, which seems unlikely because the fungal infection doesn't seem likely to result in a positive mental health outlook for a child. However, in both groups, we see that the behavioral concerns show potential correlation with each other, which is expected. In the COVID-19 positive groups, we also see potential correlations between Candida albicans and total behavioral groups, behavioral problems, depression, and anxiety. I emphasize that we're really exploring COVID-19 and how that correlates with mental health. However, we use these GI biomarkers and neurological biomarkers to detect inflammation. My discussion points in my project include the fact that patients with COVID-19 positivity have greater concentrations of antibodies to Candida albicans, and possible reasons for this include the fact that people are given more antibiotics during COVID-19, which is a precipitator for fungal infection, and also the stress response of COVID-19 or other factors might predispose children to conditions conducive to fungal overgrowth in the body's microbiome. Our other biomarkers are thus far inconclusive, and that really emphasizes the fact that it is important that this is a longitudinal study because it may be too early to detect inflammation via these biomarkers. Nonetheless, our results are strongly indicative of an imbalanced microbiome in this group of children. The limitations of my research include the fact that it is a longitudinal study and we require 3 to 6 month and 1 year follow-ups. And also we need large sample sizes to consider this investigation meaningful. And right now we have a smaller sample size of COVID-19 negative patients. Finally, we have a lot of points for comparison. We have neurological biomarkers, mental health biomarkers, and demographic analyses, and that requires complex statistical analysis. My future directions for next summers include to continue to compare the data presented to demographic factors and behavioral testing. I also have more ELISAs to do, including MMP9, gliadin, EBV, gliofibrillary acidic protein, neural cell adhesion molecule, and tau, and many more neurological biomarkers. And I want to collect gut microbiome specimens to assess the healthy or unhealthy states of the gastrointestinal community. And finally, I would love to learn the skills of Stata and Western blot to employ by myself. I would like to thank everyone for an amazing summer, and I look forward to coming again. I thank Dr. Emily Severance for all of her support throughout the summer, and a special thanks to Dr. Brown, Dr. Kampalala, Ms. Heather Thomas, and my references are accessible via this QR code. Thank you. Are there any questions? Thank you.
Okay, next up, we have a virtual presentation from Noor Huma, excuse me. Noor, are you ready?
Yeah.
Excuse me. Noor, we're gonna go right after Elenia. Elenia will be our next speaker, excuse me.
Okay, thank you, Dr. Sensei. All right, good morning. My name is Elenia Correa. This is my second summer working for the Lauro Lab for the Johns Hopkins Neuroscience Scholars Program. My research focused in the structure function correlation in the auditory system after impact acceleration traumatic brain injury in CBA-CAJ mice. To start off, I'm going to define what a traumatic brain injury, TBI, is. It is a form of acquired brain injury that occurs when a sudden trauma, such as a hit in the head, causes damage to the brain. This leads to a diffuse axonal injury caused by shaking or a strong rotation of the head by physical forces, which causes the nerve fibers to tear. This is modeled in the auditory system, which is comprised by the peripheral, the ear, and the central, the brain. Here is a general auditory pathway in which we observe the nuclei of the dorsal cochlear nucleus, the superior olivary complex, the inferior colliculus, and the lateral lemniscus, as shown here in this crescent violet sections. Now, what connects the TBI with the auditory system? We know that it leads to diffuse external injuries as well as a neurodegeneration in the brainstem. Well, it also leads to permanent sensory neural hearing loss in 50% of adults and 40% of adolescents. It also causes auditory processing difficulties. My objective then was to identify auditory functional deficits and structural damage to the auditory brainstem caused by impact acceleration TBI, IA-TBI, to show structure-function correlation. Hence, my hypothesis is that 3 days after an IA-TBI, there will be a correlation between neurodegeneration in the auditory brainstem and changes in the auditory function. Now, we model this in 3 ways. The first one is an auditory brainstem response, an ABR. This is a minimal invasive procedure done to an anesthetized mouse in which we present sounds to see if there's any neural response. On the left, you're going to see a stereotypical ABR done to a wild type mouse. Now, we extract data from this, such as wave amplitudes from these 5 particular waves and thresholds. Now, in comparison, you can see a deaf mouse, it has no neural response whatsoever, hence it doesn't have any waves. For our second method, we do have to ask a question. How can we give a mouse a TBI? What we do is that we first want to replicate what a human being would have after a whiplash in a car crash. So we anesthetize the mouse, and after that, we drop a 50-gram weight at an 85-centimeter height on its head. This will cause the head of the mouse to go down and up very quickly, hence the acceleration part of this model. After this, we silver stain. On screen, you're going to see a 50-micron thick coronal section from an IA-TBI mouse 3 days after injury. It is mostly focused on the region lateral lemniscus. So if we amplify that area, first on a sham, you're going to see the cell bodies of the region, but in a TBI, we observe silver deposits. Now, this is important because we know that as nerve fibers degenerate, they become highly agarinophilic, which means that the silver stain becomes a marker for neurodegeneration. Now, after we gather this information, we look at ABR wave analysis. So on the left side, we have a sham. The right side, we have a TBI. We have a y-axis for both graphs of the level in decibels from 10 to 80, and an x-axis of time in milliseconds. So if we gather this, in the sham, you're going to see again those 5 waves that we spoke about in the stereotypical ABR. But in the TBI, there is a slight decrease, mainly in wave 2 and in wave 4. If we look at them individually, mainly in wave 4, we do see, again, a sham, a TBI, and now we have a y-axis of amplitude and x-axis of decibel, but at baseline, which is a black marker, and a 3-day, which is a red marker, there is a trend for reduction. However, this was deemed non-statistically different. If now we look into the threshold analysis, and for context, a threshold is the quietest intensity in which the mouse auditory system is reacting to the sounds. So again, sham, TBI, but now we have the thresholds in our y-axis and the frequencies in their x-axis. We see here that there is no significant change across baseline, or black marker, and the 3 days, the red marker, mainly because we see that the error bars are overlapping each other. Next, we did a qualitative analysis of our silver stain sections. As a reference, here is what we're looking for if there is silver in our regions. And we have a y-axis of the number of sections with silver, as well as the regions of interest in the x-axis. We did have a positive control, which was the cerebellum, which was statistically significant. And even though the cerebellum, it is not an auditory region, it was still, there was a significant change, which means 2 things. 1, that the injury is effective, and 2, that the stain did work. As for our auditory regions, we did find significant change, mainly in the cochlear nucleus, the lateral lemniscus, and the inferior colliculus. Now, going back to our hypothesis, we're looking for that correlation between structure and function. And the way that we did this is by studying the amplitude and the silver. So on the left graph, you're going to see the cochlear nucleus in relationship with wave 2. And in the right side, the lateral lemniscus as it projects with the inferior colliculus in relation with wave 4. For both graphs, we have a y-axis of the amplitude and an x-axis of the number of sections with silver. What we notice here is that as you increase the silver, there is a decrease in the amplitude, and it's more emphasized on the left graph with the cochlear nucleus. So we can conclude that even though there's minimal non-significant ABR threshold increase, as well as a non-significant decrease in ABR wave amplitude, there is a significant change of silver after an IA-TBI, which brings us back to that correlation structure function, in that as you increase the amount of silver, you are decreasing also the ABR wave amplitude. Here are the references for my project, as well as I would like to acknowledge Dr. Amanda Brown, Heather Thomas, Dr. Ascenta, and Dr. Tilak, as well as my Lauer lab, Dr. Amanda Lauer, Keely Burke, Grace Capshaw, and of course, all the postgrads and undergrads working there. And finally, but not least, the mice in this project. Thank you so much for listening. Here's my contact information, and I'll answer any questions. Brandon.
I have a question.
One
Um,
Dr. Tilak, I have a question in person. Sorry, I'll be right with you.
okay. But my question is, did you have,
Yes,
did you
I'll
find a
repeat the
similar
question.
finding in the human brain, human people, in the human population?
Dr. Ratnather, we have an in-person question first, and then we'll get to you afterwards.
Okay.
All right, so to answer Brendan's question, which was why after 3 days and okay, why wait 3 days as opposed to wait more? So actually, this is the first time we're looking into 3 days. We've done 7 and up to 90 days. The reason why we're doing this is that hopefully in future directions for the project, we're hoping to find a timeline in which hearing loss or the TBI is having its effect. And the way that we do this is that we also have to check at early points, so we can start that to that timeline as well. But yeah, we already did this. This is mainly on 3 days. We wanted to see what was going on. And Dr. Tilak.
Okay. My question was, did you see similar finding in the human population? I mean, have you looked at the ABR of the human population with TBI?
Mm-hmm. Yes, so we did have stats that it's in 50% of adults and 40% of adolescents. Those are the statistics in which they're presenting permanent sensory neural hearing loss after the TBI. Was that question?
Yes.
Okay. Yes. So what I'm gonna do, we know at 3 days, there's a structure function correlation. However, as you saw in the thresholds and in the wave amplitudes, there's still no functional deficit, or there's a slight bit of functional deficit. But we do know that at 7 days, their hearing is the thresholds, which as a reference, the lower you go, the better the hearing, the higher you go, the worse. So we did see in 7 days they have worse thresholds. So that's why we're focused on 3 days to see what is going on, and again, to finding that correlation. Thank you. Thank you.
All right, so now I'm back on the correct schedule. We're gonna have Noor Huma present virtually. Whenever you're ready, Noor, you can go ahead and put up your slides.
All right. Is it not in the folder?
Oh, yeah, I believe we have it, but it might be easier. Okay. 1 second, let me make sure you have access to control the slides.
Okay.
All right. Whenever you're ready.
Wonderful. All right. Good morning, everyone. My name is Narhuma, and I'm a senior at the University of Maryland, Baltimore County, majoring neuroscience. And this summer, I've had the privilege of working with Dr. Kelly Mills and Dr. Yannick Brassoon in understanding movement disorders, specifically Parkinson's disease, and the different treatments associated with it. Our ongoing project is titled Amide Proton Transfer, or APT imaging, of protein aggregation in Parkinson's disease. Right. So Parkinson's disease is a neurodegenerative condition of the central nervous system characterized by its hallmark motor symptoms of bradykinesia, which is slower movement, and tremor. According to the Parkinson's Disease Foundation, 1 million Americans are living with Parkinson's disease, and approximately 60,000 Americans are diagnosed with it each year. Now, Parkinson's disease involves a buildup of the protein alpha-synuclein, leading to a multifaceted neurodegenerative process in the human brain. Right now, we don't have the imaging methods that can specifically show this protein buildup. And as new treatments target this protein and how they're being tested, we need imaging methods to help choose the right patients for treatment and monitor their progress. This study will test how well a new imaging technique called APT can detect this protein both in lab settings, in vitro, and in actual patients in vivo. So PD is a prevalent neurodegenerative disorder, yet early diagnosis remains challenging. The findings of structural and functional magnetic resonance imaging, or MRI, don't adequately reflect our biological understanding of relevant disease mechanisms. And dopamine transporter scintigraphy, or DAT scan, on the other hand, is considered the gold standard of supportive imaging diagnostics, but it's not widely available, and it may lead to increased individual radiation exposure in the context of a longitudinal application. What's more is DAT scan is not specific to alpha-syn pathology and shows nigrostriatal degeneration from any cause. So tauopathy, head injury, vascular disease, to name a few. Okay. So a standardized, non-invasive, pathophysiologically oriented neuroimaging method is needed for the timely diagnosis and personalized treatment of Parkinson's disease. The disease's complex pathophysiology involves multiple molecular mechanisms, which culminate in dopaminergic neuron loss due to the deposition of alpha-syn aggregates. Alpha-syn, which is found in synapses and neurons, plays a vital role in neurotransmitter regulation. Internal and external factors can disrupt the balance of alpha-syn, leading to pathological aggregation. Addressing these mechanisms could pave the way for more effective therapeutic strategies to tackle PD's progression. We propose a method that may be suitable for imaging alpha-syn content and aggregation non-invasively in the human brain. As a new MRI contrast, chemical exchange saturation transfer imaging, or CEST, allows the detection and visualization of specific molecules in biological tissues. It is a form of contrast-enhanced MRI that relies on the exchange of protons between different molecules, and it offers far-reaching potential for molecular imaging in neurodegenerative diseases. Right, so amide proton transfer, APT imaging, a type of CEST, can detect protein aggregates and differentiate PD patients from healthy subjects. It offers potential for individual disease monitoring, paving the way for individualized therapies and identification of at-risk individuals. It allows the measurement of mobile peptides and proteins both in vitro and in vivo, sensitively detecting protein aggregates related to PD pathology. It has already shown diagnostic utility in differentiating PD patients from healthy controls, and it has the potential to serve as a surrogate marker for alpha-syn pathology in vivo, aiding in disease monitoring and personalized treatment strategies. It could also help identify at-risk individuals who could benefit from pre-symptomatic neuroprotective treatments. All right. So this study aims to evaluate the sensitivity of APT imaging contrast in Parkinson's disease, which focuses on detecting aggregated alpha-syn in vivo. And so those main objectives are to assess APT imaging contrast for detecting aggregated alpha-syn using physiological phantom studies, then comparing APT imaging in persons with Parkinson's disease, so PWPD, to healthy controls, HC, to identify differences. And then correlating APT imaging with cerebrospinal fluid or CSF markers of alpha-syn pathology in patients with Parkinson's disease, including an alpha-syn level, PY39 alpha-syn, and RT-QuIC, or real-time quicking induced conversion, used for the detection and amplification of abnormal proteins, particularly prions, or PMCA aggregation assays, along with other relevant biomarkers. All right, so our methods, just an overview, recruitment, and then obtaining consent, initiating a baseline study, the MRI, biofluid sampling, so the blood draw, and the optional lumbar puncture, and then analyzing. All right, so persons with Parkinson's disorder or PWPD will be recruited from Johns Hopkins, the Parkinson's Disease and Movement Disorder Center, under the direction of my PI, Dr. Mills, which are either approached directly or they're referred to us by other physicians at our center. Healthy controls are then recruited from the families of the individuals with Parkinson's disease during a regularly scheduled clinical visits with Dr. Mills. And alternatively, if they are a spouse or family member of a person with Parkinson's disease, seeing someone else in the center, they can express their interest, that physician, and will be referred to my PI, Dr. Mills, or Dr. Prasoon. Then potential participants receive the informed consent form ahead of the consenting/baseline study visit. The consent process will take place at the beginning of the study visit, and then written documentation of consent is obtained. All right, the baseline visit. So both patients with Parkinson's disease and healthy controls have the baseline study visit that lasts between 2 to 4 hours, where the following procedures are performed. So we evaluate medication history, stat coagulation studies, platelet count, medical history, the Movement Disorder Society's clinical diagnostic criteria for Parkinson's disease, reviewing inclusion and exclusion criteria, olfactory testing, which is optional, the MDS-UPDRS, which is the Movement Disorder Society's Unified Parkinson's Disease Rating Scale, the Beck Depression Inventory, the Montreal Cognitive Assessment, also known as MoCA, and then essentially the MRI screening form. All right, so the MRI procedure, approximately 60 minutes, no contrast agent is used. Volunteers are asked to lie still in the magnet, provided with foam padding for added stability, and earplugs to reduce noise. And 2 types of MRI measures are obtained: the anatomic information of the brain, so T1 weighted and FLAIR weighted MRI, 7 minutes. Then APT contrasted imaging sequence, that's 30 minutes for protein and pH information. Okay, so biofluid sampling. The blood draw, we have certified staff that collect approximately 9 milliliters of venous blood during the baseline visit for analysis of alpha-syn levels, alpha-syn aggregation measured by RT-QuIC or PMCA, and other potential biomarkers like PY39 alpha-syn. Then the optional lumbar puncture. So they can go under the lumbar puncture that's conducted by a qualified neurologist or nurse, and up to 27 ml of cerebrospinal fluid is collected. The first 2 to 5 ml is analyzed immediately for cell count, protein, and glucose levels. And then Dr. Mills will then review the abnormal results for clinical significance. And participants will also be informed if there are any clinically meaningful abnormal values, and they'll be then referred to appropriate physicians. Okay, so the sample analysis. So serum undergoes testing for alpha-syn concentration and strain type with RT-QuIC or PMCA aggregation assays. Some assays may include enrichment for exosomes, including LCAM1 enriched exosomes. Other candidate biomarkers for PD progression, such as PY39 alpha-syn, informed by parallel preclinical studies, will also be evaluated in the serum samples. Then the CSF, if completed, undergoes testing for alpha-syn levels as well, and aggregation with RT-QuIC or PMCA, similarly, and other candidate biomarkers for PD progression, such as the neurofilament light chain. All right. So our study will provide substantial benefits to the diagnostic evaluation of patients with Parkinson's disease. APT imaging has potential applications in Parkinson's disease, and as it has with other neurodegenerative diseases, such as assessing early stages of the condition, stratifying patients for clinical trials, and monitoring individual disease progression and therapy. right, finally, altogether, APT imaging can pave the road to generally individualized medicine through non-invasive neuroimaging. Our goal is to ultimately develop a non-invasive tool to assess brain function and identify alpha-syn aggregation, the hallmark of Parkinson's disease. And successful APT imaging could lead to improved diagnosis, patient stratification, clinical trials, and individualized therapy monitoring, ultimately advancing our understanding of the disease and facilitating personalized treatment approaches. I'd love to give a warm thank you to the following: Dr. Mills, Dr. Yannick Brassoon, Dr. Amanda Brown, Dr. Tilak Ratnanathar, Ms. Heather Thomas, Dr. Sante, Dr. Claire Carter, and all of my peers in the JHNSP and JHNeurophytes programs. Thank you all so much. And these are my references. Thank you so much. Any questions?
No, I don't see any. Great job, Nora. Thank you. All right, next up, we have Sophia Spindler-Craig. Slides up.
Great. Hi there. My name is Sophia Spindler-Craig, and I'm part of cohort 5, and I'm a rising freshman at Grinnell College, hoping to study biochemistry, but I'm from Rochester, Minnesota. This summer, I had the opportunity to work in Dr. Solange Brown's lab, mentored by her and then Dr. Ahmad Taha. One of the projects that I've been working on is the one that I'll present today, focuses on pupil size as a proxy for reward history and in a dynamic decision-making task. Decision-making in those with neuropsychiatric diseases can be impaired, causing them to make riskier and possibly unhealthy decisions for them. We make decisions every day, of course. It could be the kind of food we choose to eat, nutrient-dense foods for long-term health, or sugary, high-caloric foods for instant gratification. We weigh the reward for both choices and ask the question, which one do I choose? A question that becomes more and more important as we make our way to medical professions, interacting with patients and making quick decisions for our future patients and our research. In this previous study, a mouse's behavioral state can affect pupil size. Pupil diameter in this graph reflects the state of the animal. This graph then shows the pupil dilates more if the outcome is sure or has low uncertainty, which is shown in light gray. It also shows that for high uncertainty trials in black, the pupil dilates less. Based on these graphs, we can infer that pupil size can be dependent on the mouse's behavioral state. In the task that we give the mice begin by being presented with 2 lick ports. Each lick port has a probability of giving the mouse a reward. For example, in this trial, one could be 50-50, but in another trial, it could be 10-50. These probabilities mean that the mouse is choosing where to lick dynamically. Depending on the previous rewards, the mouse has to guess which side to lick. Prior to each one of the trials, there's a 1-second period that we call the no-lick window, where we ask the mice to not lick either port. We do this to minimize the amount of spontaneous licking, which could skew our results if not controlled for. That way, we know the mouse licks as a response to the conditioned stimulus, which is a tone that signals to the mice to choose the port that they think will give them a reward, each with a continuously changing probability, as I mentioned earlier, to give a reward to the mouse. The mouse's choice reflects what it thinks is going to be the highest reward port. This graph shows when the mouse alternated ports during the session, based on changing reward probabilities. The dark gray lines are trials rewarded, and the light gray are trials unrewarded. That also includes the probability of reward on each port, whether it be 10-10, 50-90, 90-90, or others. Using the frames of the video during the no-lick window, we used a deep neural network, which is called deep lab cut, to mark each side of the pupil and track the coordinates of both markers. Here we show 10 seconds of the video deep lab cut gives us after training the network. As you can see, deep lab cut is an accurate and reliable way to measure pupil size. We then calculated the circumference of the pupil by taking the difference between each marker and multiplying that by pi using Python and MATLAB. We also found reward history, which takes the past 10 trials and calculates the number of rewards. It is calculated as a sliding moving average, which means recent rewards like trial 10 are weighted much higher than trials 1, 2, and 3 during the calculation. This here is an example of high reward history, where the dark blue boxes are trials where the mouse was rewarded, whereas this would be a low reward history. We hypothesized that during the no-lick window, the mouse pupil would be more dilated at a high reward history, whereas it would be less dilated at a low reward history. Instead, we found this. Here we show pupil size as a function of reward history. Since we graph this as a linear correlation graph and the slope is pretty much near 0, we can infer that the pupil size and reward history are indeed not correlated. We then made an alternative hypothesis. We hypothesized that in a high reward state, the pupil is more dynamic, and in a low reward state, the pupil is less dynamic. We call this pupil reactivity, which is calculated by taking the change in the pupil diameter during the no-lick window. When studying our alternative hypothesis, we can see that the slope of the line is more significant than in the previous graph. With this graph, we can infer that there is in fact a correlation between pupil reactivity and reward history. From our research, we can conclude that pupil size is not correlated with reward history, but pupil reactivity is correlated. We are interested in researching further to find whether there are differences across mice, or if the pupil dynamics are a general feature of pupil reactivity reflecting reward history, and determining how pupil reactivity might change as the mice learn the task. I want to acknowledge Dr. Solange Brown, Dr. Ahmad Taha, Dr. Amanda Brown, Heather Thomas, Dr. Salik. But then additionally, I would like to thank Dr. Sante, the other scholars, and especially the rest of the Brown Lab, including the other undergrads, Jenny, Alec, and Sam Bronkers. Thank you. Any questions? No? Yeah, so the question was why I was recording the pupil size during the no-lick window. And the reason that I did that was because we wanted to see if there was like either something to do with anticipation of the reward that the mouse was getting. So as they get closer to getting the reward, maybe their anticipation grows, which could mean that their pupil size grows because anticipation often goes along with arousal, which I mentioned earlier. If somebody is more aroused, then their pupil size is going to grow. It's also a way to externally see if the mouse maybe understands a task before applying that knowledge. So since there's been a proven understanding that there is like a difference in time between a mouse understanding the task and then being able to apply the knowledge and actually correctly completing a task, we wanted to see if pupil size could be a way that we can see if the mouse understands the task. Thank you.
All right, we're going to take another break. It is now 11:16. If we could all please return back to the Zoom room by 11:20. 25. And I would also encourage you, I'm going to put the slideshow back up, but please, all of you who are attending virtually, please take the time to review all of our posters. All of our scholars did a lot of really hard work on them. And please also remember to use the survey to judge each of the posters that you look at over the course of the day. We'll be giving out awards to the best posters and best podium presentations at the end of our program today. And Dr. Brown has a couple words for us as well.
Thank you so much, Santi. I just wanted to say welcome to everybody. Thank you so much for joining. We had a fantastic morning and we look forward to having a fantastic afternoon. So please do come back and join us and celebrate our outstanding scholars. Thank you all.
in just a second.
So then keep these muted, right?
Yes, mute yours. All right, thank you all for coming back and joining us again. We're going to be starting up with our next block of presenters. Up next is Luis Guzman Clavel, and he's going to be our next speaker. Go ahead.
Hi, everyone. My name is Luis Guzman, and I'm part of the colloquial lab, and I'll be sharing with you today part of my summer research this year, which was about neuronal connectivity in general, and more specifically about MEF2C and how loss of MEF2C leads to deficits in innervation of layer 2-3 neurons. But before we start speaking about neurons and connectivity, we can take a step back and start with the mouse brain. Not as complex as a human brain, but very structurally similar. And if we take a coronal cut and then look at a coronal plane, we can start looking at structures, for instance, like the neocortex, which is very important for high order behavior. And it's composed of multiple parts like motor cortex, sensory cortex, visual cortex, to name a few, but our main focus today will be in primary somatosensory cortex. So the neurons that we'll be speaking of are part of this area of the cortex. And in particular, it's let's give a primary cortical development, which happens in an inside-out manner, meaning that there is a population of cells that coordinate together to arrange this inside-out fashion development, in which lower cortical layers start migrating upwards first, and then we have higher layers at later times in embryonic development. For instance, layer 6, then layer 5, then layer 4, and then layer 3 at E15.5 or embryonic day 15.5, and finally after birth, postnatal development. And the importance of this layered structure, for instance, one of the important features of this is that it allows for a very specific population grouping that later on ends up in very specific connectivity patterns. For instance, after postnatal development, we can see very specific and stereotyped patterns of connectivity in each of the layers. For instance, layer 2-3 neurons branch out their axonal collaterals only in layer 2-3 and layer 5, but not only in layer 2-3 and layer 5, but not in layer 4, for instance. And this pattern of local axonal collateral extension is also joined by long-range axonal projections. Sorry about that. It's also joined by long-range axonal projections that have also a very high degree of specificity. And after seeing that these very highly specific connectivity patterns arise, it begs the question. It begs the question of, well, how do these neural connectivity patterns arise so specifically? What drives connectivity to be so specific? And so there's a lot of abbreviations there. Bear with me. We'll explore them together in a second. But these are just canonical innervation areas of layer 2-3 neurons. And one of the questions that we can rephrase these as is like, why don't layer 2-3 axons project like this? Then why is it not a general cortical innervation? Or why don't they project like this, only to one particular spot? And well, one of the reasons for this is that in the extracellular environment, there are actually guidance cues or attractive or repulsive cues that dictate whether the axon will develop in that local area or whether the growth cone will extend towards that particular neural brain area. And the same goes for long-range projections as well. And of course, this has to happen with extracellular guidance cues, but the neuron has to have the ability to respond to these extracellular cues. And the way that it does that is that it has very specific expression of proteins that allow for this. And for instance, after developmental neuronal activity-induced transcriptional changes, which is a mouthful, but only means that after developmental neuronal activity, not synaptic activity, we have transcriptional change regulation by transcription factors, for instance, MEF2C, which then leads to proper expression of receptor molecules, cytoskeletal proteins and such. And finally, this appropriate protein expression leads to the axonal branching or the axonal extension in the appropriate direction. And so in summary, to get from this to that environment in which we can accurately react to the cues outside, there need to be appropriate receptors, so adhesion molecules and cytoskeletal proteins like trebrin, which are in turn regulated in their expression by transcription factors, in particular MEF2C. And so MEF2C is very highly expressed in layer 2-3 neurons and in the cortex as well. And as you can see there in that in situ image from the Allen Brain Institute. And it is also known to regulate cortical neuromigration, maturation, and neural development. And after activity occurs, it goes into the nucleus where it, of course, like most transcription factors, regulates the expression of other genes. And this leads to normal brain development. But upon loss of MEF2C, we actually observe a wide array of neurodevelopmental disorders like ASD, schizophrenia, including MEF2C haploinsufficiency syndrome. And so what we have in this case is we have a conditional knockout mice, which we can induce after electroporating Cree at E15.5. And the reason we do it at E15.5, of course, is because layer 2-3 neurons are migrating, the neural progenitors of those neurons are migrating upwards at this age. So we can specifically target this region and only selectively deliver the DNA to these neurons. Afterwards, this is followed by perfusion and brain isolation at P14 mostly, and also at P28. And then eventually immunohistochemistry, and then imaging and analysis of the brain slices. And so first we'll talk about the long range, projections and the phenotypes that we observe with the MEF2C conditional knockout mice. And we'll start with the S1, S2 border innervation. As you can see, there's innervation both in the ipsilateral or same side and the contralateral side of the neurons. And what we can see here is that in the ipsilateral S1, S2 border, you can observe very high, very prominent innervation, but then in the conditional knockout mice, you can observe that this innervation is abolished. Similarly, in the contralateral region of this type, we can see the same, where we have two columns of innervation, one in the S1, S2 border, and then another one in the secondary somatosensory cortex. And then we also see the loss of this innervation, which was quantified by fluorescence intensity normalized to the neutral electroporation efficiency. And similarly, if we move over to the insular cortex, which is another one of the canonical innervation areas, what we can observe is that the MEF2C conditional knockout decreases innervation to the insular cortex. So in the control mice, we have this ipsilateral highly innervated area in the ipsilateral side, and then in the upper part of that innervation, we can see a loss of innervation. Similarly, in the contralateral side, we see a similar defect, as well quantified by fluorescence intensity. And then another one of these interesting phenotypes is that of what we observe across the corpus callosum in the motor cortex. So of course, these layer 2-3 neurons are callosal projection neurons, which means that they project their axons to the contralateral side of the brain through the corpus callosum. And then they also innervate this area right here, the motor cortex. And what we observed is that in the conditional knockout mice, as you go across the corpus callosum, you can see that there is a loss of fluorescence intensity. So in the control, you can see that there is a very stable fluorescence signal, but then in the conditional knockout, we can see that it starts to diminish after midline crossing. And we divided this into 3 areas, which we later quantified specifically. And we can see that after the midline, there is a loss of fluorescence intensity. And something that we observed parallel to this was that in the motor cortex area, which is quite close, like as you can see here, to the corpus callosum in the midline, is that normally this area is innervated properly. However, in the conditional knockout mice, what you can see is that we see this looping rather than it going upwards to innervate the upper layers. And in the contralateral side as well, even more prominently, you can see that instead of innervating these upper cortical layers, it simply loops back. And finally, to make sure that this was not a developmental only phenotype that could be later corrected, we also repeated the experiments at pin 28, and we observed the same phenotypes and similar features in the conditional knockout brains. And something that we had mentioned before was also that there are also local axonal collateral branches that are very important for the connectivity and the function of these cortical areas, not only long-range projections. And so we also looked at the local axonal collateral phenotypes to see if there was any change in the conditional knockout mice. We did this through a sparse labeling technique, where we can selectively tag only a few neurons in the brain rather than the high amount that we do in the bulk electroporations. And what we look for is these axonal collaterals that are not supposed to be there in layer 4, or a decrease in the normal layers where there should be innervation. And what we observed was that there was a decrease in the primary branches in layer 2-3 in the homozygous mutant, and then an increase in ectopic axonal collaterals in layer 4 in the heterozygous mutant, which are not normally supposed to be there. So there was an increase in branching in the area that normally does not have any collaterals. And so just to recapitulate the phenotypes, in both secondary somatosensory cortices as well as in both insular cortices, we can observe that there is a decrease in innervation as well as this ectopic looping in motor cortex. And then finally, we can also see that there are local axonal collateral phenotypes that are both decreasing in layer 2-3, where normally there will be more innervation, and then increasing in layer 4. And so this leads to a model where loss of activity-induced transcriptional changes that are normally regulated by MEF2C lead to the inappropriate expression of the molecules that we had previously mentioned, and therefore inappropriate extension or branching in response to the guidance cues in the environment. And so just as future directions, we can manipulate the activity of layer 2-3 neurons with a paradigm that we have characterized now in DREADS, which we can use to excite neurons or inhibit neurons, but we also are using KRA2.1, which is an inward rectifier potassium channel that leads to inhibition. And then we have seen similar phenotypes as MEF2C, so we will try to rescue this with overexpression of MEF2C. And then there is also an available single cell RNA sequencing database, which we can look for downstream targets of the transcription factor there. We're very interested in ephrin and EPH because they have area-specific expression in these specific target areas of layer 2 neurons, as well as we can also do brain clearing and then light sheet imaging, where we can do 3D renders and see the innervation more in a more high detail manner. And finally, just testing whether their phenotypes arise from an activator or repressor role in F2C. So thank you so much for your attention. And if there are any questions, I'll be happy to take them. First, for the acknowledgements, I would like to thank Dr. Kolutkin for taking me into his lab, as well as Sriram for working closely with me and mentoring me. And of course, the team, Ms. Heather Thomas, Dr. Amanda Brown, and Dr. Asante, as well as the rest of the scholars. Thank you for a great summer. Oh, yeah. So Dr. Amanda Brown was asking about the conditional knockout that allows us to also manipulate it in a very temporally controlled fashion. And the conditional knockout was previously characterized, and it allows us to, it's a Cre-dependent conditional knockout model in which we can neutral electroporate or deliver a Cre by electroporation or by another method of DNA delivery. And then in only the Cre-expressing cells will there be the knockout that we observe. So it's not a full brain knockout or not a full animal's knockout, because actually those are lethal of MEF2C, but then the conditional knockout only in layer 2 neurons that we selectively target. Thank you.
All right, our next speaker will be virtual. It's going to be Charles Pinto. Charles, are you on the call? Oh, you're here? Oh, okay, then we don't need to do it virtual. He's here with us in the room. Let me just get his slides set up. Yes, the captions are up.
right.
Good morning, everyone. So good morning, everyone. My name is Charles Pinto, and this summer I've been investigating the role of the human ApoE4 haplotype on adulthood outcomes following neonatal hypoxia in mice with the Chavez-Valdez laboratory. So our lab is a neonatal lab, which means that our research is focused on events that occur during the neonatal period, which is the first 4 weeks of a newborn's life. So we're specifically interested in neonatal hypoxia, which is the single most common brain injury in full-term neonates. Now, neonatal HI is a brain injury that prevents adequate blood flow to the infant's brain as a result of a hypoxic ischemic event during the prenatal, intraperitum, or postnatal period. Hypoxia refers to a state of insufficient oxygen, while ischemia refers to a state of inadequate blood flow. Now, the unique approach our project is taking is that we're interested in understanding the impact of human apolipoproteins in the context of neonatal HI. So apolipoproteins are proteins that combine with lipids to form lipoproteins. And we are focusing specifically on the apo class E proteins, which have been implicated in mechanisms related to aging, dementia, and Alzheimer's disease. Now, there are 3 characterized human ApoE alleles. We've got ApoE2, ApoE3, and ApoE4. ApoE2 is protective against Alzheimer's disease. ApoE3 is neutral, while ApoE4 presents the strongest genetic risk factor for Alzheimer's disease. Now, given that in healthy children, that same ApoE4 allele is also associated with reduced temporal cortex thickness and hippocampal volumes, which are in turn correlated with reduced spatial memory, we're interested in how the ApoE4 haplotype may modulate responses to neonatal HI. So with this in mind, we hypothesize that the ApoE4 allele exacerbates early neurodegeneration and results in lasting neuroinflammation, ultimately leading to behavioral impairment 6 months after neonatal HI. Now, in order to test this hypothesis, we induced a cerebral HI injury at P10 using the Benucci model, which consists of a right carotid ligation followed by 45 minutes of hypoxia in order to simulate a hypoxic ischemic event. From there, between 10 days and 6 months of age, we conducted various forms of neurobehavioral testing. At 6 months, we conduct experiments on both fresh tissue and 50-micron brain slices in order to measure levels of astrocytosis, neuroinflammation, oxidative stress, and neurodegeneration. Now, in conducting these experiments, we see some really interesting results. First thing that we see in these pie charts over here, is that HI mortality is doubled in mice with either ApoE allele. So a rate of 39.2% for the ApoE3 mice and a rate of 43.9% for the ApoE4 mice. What's interesting though, is that the ApoE4 mice tend to die sooner during hypoxia than their wild type or ApoE3 counterparts, which you can see in this bottom left graph here. And the ApoE4 mice tend to have a 4-fold larger seizure burden than the wild type and ApoE3 counterparts, which you can see in this graph over here on the right, which basically means that these mice are seizing for longer periods of time. Now, the differences between the ApoE3 and ApoE4 mice can also be seen in the brains themselves in the bottom right here, where we see that the ApoE4 brains display a greater degree of inflammation 6 months after the initial HI injury. So with regards to behavioral differences, we see that ApoE4 mice spend both more time in the periphery and travel greater distances of the open field assessment area. So I think these heat maps and tracking plots at the bottom here are really interesting and a great visualization of that. A great visualization of that because we can see in this farthest most tracking plot on the right that the ApoE4 HI mice spend very little time in the center of this plot right here. They're spending all their time traveling in the periphery, which is representative of anxiety-like behavior in these mice, because we find that they're hesitant to venture beyond this periphery of the assessment area. So with all this in mind, the question becomes why, right? We see these differences between these ApoE3 and ApoE4 mice, but what are the cellular bases for these differences? So So in order to answer that question, we started by looking at markers of neuroinflammation, namely markers of mainly levels of NF-kappa B and pro-inflammatory cytokines TNF alpha and interleukin-1 beta. So the way that these graphs are set up is that we have hippocampus and temporal cortex levels on the top and bottom, and then the sham mice are represented by the green boxes, and the injured injured mice are represented by the blue boxes. And what we see is that 6 months after injury, the hippocampus and temporal cortex of the injured APOE44 mice display increased levels of NF-kappa B, as well as increased levels of both pro-inflammatory cytokines of interest. Moreover, 3-nitrotyrosine, which we see in this farthest graph on the right here, which is a biomarker of protein nitration and nitrooxidative stress, which is associated with free radical protein damage and impaired astrocytic function, was also increased in the hippocampus and temporal cortex of these injured APOE44 mice compared to the wild type and APOE33 mice. So the next thing we looked at was cytosolic accumulation of phosphorylated tau, or p-tau. So p-tau is a common pathological feature of neurodegeneration and is thus an interesting point of difference between these APOE3 and APOE4 mice. So using IMARIS, which is an image analysis software, we analyzed more than 7,500 cells. We found that 501 of these cells express cytosolic p-tau, which in this graph on the left are labeled as p-tau positive cells in red. So what this graph tells us is that irrespective of genotype, increased cytosolic p-tau occurs in cells with pycnotic or shrunken nuclei, which itself is an indicator of cell death. Now, the IMARIS rendering on the right allows us to see that these pycnotic nuclei from p-tau positive cells are stained with DAPI and marked with a green asterisk right here in the bottom right corner, while the nuclei of the phospho-tau negative cells are represented by these hollow spheres and marked with a blue asterisk here. So these are all phospho-tau negative cells. We can clearly see that the DAPI stained nuclei are smaller, even with the accumulated phospho-tau sections in red, than the hollow or healthier nuclei. Now, the reason why this is significant is because phospho-tau cells are also markers of neurodegeneration. So the increased number of phospho-tau positive cells with pycnotic nuclei in the CA1 and CA3 regions of the hippocampus, the injured APOE44 mice compared to all other experimental groups is thus further evidence of their heightened neurodegenerative state. Now, the final cellular point of difference we wanted to investigate was astrocytic density and complexity. So as you can see in this graph on the left here, astrocytic density was increased in the CA1 and CA3 regions of all APOE44 mice. Now, the graph on the right shows that within the APOE44 experimental group, the complexity of astrocytes is greatest in the hippocampus of HI mice, which is represented by this blue dotted line in the top and bottom. Now, the reason why this is significant is because of the positive correlation we saw between astrocytic complexity and the p-tau positive cells we talked about earlier, graphed out in this figure on the right here. This relationship between astrocytic complexity and p-tau accumulation in APOE4 sham and APOE4 HI mice can be seen in this panel on the left, and suggests an association between astrocytic dysfunction and neurodegeneration in the HI injured APOE44 hippocampus. Now, taking these results together, we speculate that APOE4 exhibits harmful toxicity in the neonatal brain correlated with neuroinflammation, neurodegeneration, and cell death during neuronal maturation after neonatal HI. As such, APOE4 can be identified as a risk factor for long-term late life consequences resulting from neonatal HI amenable to intervention, which I think is a particularly exciting direction for our future research. So I want to give a special thanks to the entire Chavez Valdez Laboratory, Mark for mentoring me in the lab, as well as Dr. Brown and Heather for the opportunity to be here and supporting us, all the scholars, and Drew and Ben as well. And thank you all for listening. If you have to take any questions. Could you repeat the question one more time? I didn't hear part of it.
I'm sorry, just briefly.
Mm-hmm. Mm-hmm. Right.
So you're showing us
Mhm.
Start to initiate that process.
Yeah. Yeah, I mean, our research is mainly focused on kind of the overlap between this really common brain injury in neonates and APOE4 and how this kind of subgroup of the population is put at a greater risk or maybe predisposed to worse outcomes later on in life. So they're both predisposed to Alzheimer's and then maybe worsened outcomes later on. So yeah. Yes.
You mentioned towards the end about like, a bit of the future directions of this line of work. Do
Mhm.
you see this correlation with APOE4 fitting more in with improved prognostication, or do you think they could fit and kind of contribute towards the development of novel therapeutics?
I think a little bit of both. I think that right now, therapeutics for HI are very limited and they're focused more on kind of, I'm not sure what the word is, but they're focused more on treating rather than preventing. So the APOE4 kind of could fit in as an early identifier so we can treat this disease earlier. So. Okay, thank you.
Thank you, Charles. That was wonderful. Next up on our schedule is going to be for Laura Bellamy.
Hi, everyone. My name is Flora, and this summer I've been granted the privilege to work under Dr. Brown's lab and focus on NF-kappa B, which is a central pro-inflammatory modulator. And maybe you've picked it up across other people's presentations, but NF-kappa B has been mentioned. And essentially, I'll just be diving into why it is a pro-inflammatory modulator, and what work I've been doing. So before we begin, we must understand that NF-kappa B's essential role is to have dimers, and these dimers are molecular bases that go into the nucleus and create whatever genetic transcriptional change that is needed. So essentially, there are two pathways. For today's purpose, we'll be looking at the canonical pathway. And for the canonical pathway, we can, let me just do this. For the canonical pathway, there's usually a cytokine or a toll-like receptor that binds to a ligand, initiates a canonical pathway. For this purpose, we'll be looking at TNF-alpha, which is a very potent and important pro-inflammatory cytokine. And through a series of downstream signalings, we can reach the NEMO, which is our essential modulator. And essentially, their job is to attach a phosphate group. Sorry. Okay. No, this is not. Yeah. Sorry about that. Okay, yeah. So going back to what I was saying, NEMO is our essential modulator, and essentially its job is to add a phosphate group to ICPA. ICPA is the star of this show, and ICPA's job is to act as an inhibitor and hold these dimers in place from reaching the nucleus. So when NEMO is activated via the TNF-alpha, it releases a phosphate group, which essentially degrades ICPA and releases the dimers to enter the nucleus. Once it enters the nucleus, it creates transcriptional change. And in this example, we can see cytokines being released, meaning that these dimers were for pro-inflammatory responses. Understanding that, we have to understand how this relates to HIV and the positive feedback loop within the brain. So we must ask, how does this virus enter the brain? Now, just like COVID-19, HIV must infiltrate the brain via the blood-brain barrier, which is a very highly permeable, or a very highly selective membrane in which certain ions and cells can come in. Now, one of those cells can be monocytes. Monocytes can be infected by HIV and kind of be hijacked and can go through the blood-brain barrier. Now, immunologists call this a Trojan horse model. And like the city of Troy, danger is sensed by the microglia. Once the danger is sensed, the microglia, as follows, releases the cytokines via the NF-kappa B pathway that we spoke about. Now, an important one is CCL2, which drives us to recruit peripheral monocytes. Unknowingly, this happens and recruits peripheral monocytes that have been infected with HIV, increasing the population of HIV in the brain. Now, the viral protein TAT hijacks itself into the NF-kappa B pathways we spoke about. Remember ICPA, the green inhibitor? Well, the virus protein TAT degrades ICPA and prevents it from doing its job, which means that these pro-inflammatory dimers are free to go to the nucleus without anything inhibiting from it. Now, this releases a very pro-inflammatory state. And at very basal levels and at normal conditions, nitric oxide is used at minimal levels to degrade whatever debris is needed. However, in this pro-inflammatory positive feedback loop, there is a very high concentration of nitric oxide, which becomes incredibly dangerous to neurons and lead to neuronal degeneration, which is why we see HIV patients later on developing dementia, for example. Now, what did I specifically do for my methodology? Understanding this, I wanted to look at the brain tissue slices that my lab provided and better understand the role of NF-kappa B by sex. Given some literature I have read, I've understood that HIV behaves differently by the sex of people and mice, so I decided to divide them by group. Now, osteopontin is a very potent and important protein in our lab. Osteopontin is a very biologically diverse protein. It is located on our 4th chromosome. It comes from the bone marrow, but it is involved in many pathways such as NF-kappa B. Osteopontin has been found to create pro-inflammatory effects such as upregulating the NF-kappa B pathway, similar to the TAT viral protein. However, in our lab and in other labs and literature, we have found that NF-kappa B, osteopontin, can act as an anti-inflammatory protein in a protective state. So there's very little what we know about the osteopontin's very complex array of what it can do. Understanding this, I created a positive control with mice having a buffer being uninfected with osteopontin and a negative control with no osteopontin and uninfected. And I replicated that with the HIV group. When I came into lab, these mice brain tissues have already been sectioned to me and labeled M as a mouse number. For example, M34 would be mouse 34 and sections ranging from 1 to 20. And as this diagram shows, 1 being closer to the forebrain and 20 being closer to the cerebellum. Keep this in mind, this might be important. So we use IHC to better understand the fluorescence of our study. And IHC uses antibody fluorescence to tag the specific thing that we're looking for, essentially. Mine was NF-kappa B, like I spoke about. So on day 1, I would get my slide tissues, collect the necessary information, and go through a multi-series wash with xylene and a series of alcohol, and then rehydrate it with TDS. I would use proteinase K that would help better aid the necessary antibodies for coming forward. And then after that, I would then add my primary antibody dilution and incubate it overnight. The next day, I would then wash my slide and then add my secondary antibody that's tagged with the fluorescence and then incubate that for a bit. And then I would mount my slide with DAPI, which then highlights the cells blue. Given that I divided my mice by sex, I saw a very starkly different fluorescent leveling between my groups. For my female groups, and understand that I overexpose these photos for the purpose of this presentation, but for the female groups, we can see a very circular, rod-like morphology around the cells, as I've been highlighted in red. Now, understand that the limitation to this is that this is a 2D picture, so we do not understand where exactly in the cell the NF-kappa B is clustered the most. But it was more important enough for me to know that compared to the male tissues, there is overall very little fluorescence. And when there was NF-kappa B fluorescence, it was behaving next to the nucleus or within the nucleus. And given what we know about NF-kappa B and what I spoke about earlier, when it is in an activated state, it is much closer to the nucleus. So that tells me, visually speaking, that NF-kappa B is acting more of an active state, visually speaking. Now, the next step was to measure my cells. And on the graph to the left, or yeah, your left, the blue one, I wanted to make sure that the pictures or the slides that I had all had the same level of cells so that I wasn't unfairly measuring one of the groups and having incorrect data. However, the blue chart shows that these numbers of cells ranging from 250 to 400 were visually more so symmetrical, as the X represents the mean or average, and they all were just pretty much symmetrical. So that told me that was really good. However, for the fluorescence one, the y-axis being the FTSE fluorescence, which was marked in green, and the x-axis marking every single group, we can obviously see that HIV OPN plus had the highest level of fluorescence compared to buffer OPN plus, which had the lowest. Buffer OPN plus, remember being a positive control, behaved really well. However, our two OPN knockdown mice looked almost identical. Looking at this visually side by side, the buffer OPN plus showed that the female group had a more normally distributed arrays of fluorescence, whereas the male group had a left skewed distribution. And highlighted under these mice showed me that when the mice tissue was closer to the forebrain, that it would show higher levels of fluorescence versus when it was closer to the cerebellum. So that immediately told me maybe NF-kappa B is not equally distributed throughout the brain. As you can see with the knockdown counterpart at a much higher level range, or excuse me, much lower level range, that we can see a similar trend of, with the female and the males. Now going to our infected group with HIV OPN plus, remembering that high level of variance, we can see that the female group contributed to the highest amount of fluorescence versus the male group, where some of them had negative levels of expression. Now, HIV OPN knockdown was very similar in terms of distribution with the buffer group. With taking at these side by side, we can see that the female groups were both normally distributed and the male groups had a left skewed distribution. To me, that tells me that OPN has a very important role in NF-kappa B signaling, and more studies need to be done to better understand this trend. Now, next summer, I hope to stain these same tissues and groups by finding macrophages and astrocytes and see what specific cells are expressing these levels of NF-kappa B. And I want to stain for specific sections of the brain using NF-kappa B expression. For example, maybe I would stick to just one mice and collect and stain all of its brain tissues. And thankfully, we have a specific microscope that can help better aid in capturing that. And I think that would be the best help for next year. Here are my references. And I would like to give a very warm thanks to Dr. Brown for being an amazing mentor. I learned a lot and grew a lot as a person, as well as Heather, Dr. Carter, and members of the Brown Lab who've been incredibly patient and kind with me and my endeavors, as well as the scholars here. Thank you guys so much for being here. I would like to open the floor to questions. Thank you.
Very clear.
Thank you, guys.
All right, next up, we have Amy Padilla, who's going to be presenting her talk virtually. Amy, you are perfect. Whenever you're ready, go ahead and share your screen, okay?
Okay, thank you so much. Okay, okay, can everyone see my screen?
Yes, we can.
Okay, awesome. So my presentation is on the exploration of the effects of nitric oxide on apolipoprotein E function, or ApoE, in neuroinflammation. And so the focus of, or what you need to understand in order to understand the entire research, is nitric oxide. So nitric oxide is an unstable and reactive signaling molecule that exists at higher concentrations in neuroinflammation. And neuroinflammation occurs in a lot of diseases, but one that's significant in my lab that's being studied at the moment is multiple sclerosis. So, NO, because it's a signaling molecule, we know that it can change the behavior and function of cells and proteins. And this is through a process called nitrosylation. And nitrosylation is basically the attachment of an NO molecule to a protein. So when this happens, it can change the behavior function. And then little is known about the effects of nitrosylation, but must be understood because NO plays a lot of lead roles in diseases like multiple sclerosis. So in EAE, which is a mouse model, NO targets ApoE, which is what we found through experiments and is important for the functions of microglia, like conducting phagocytosis. And phagocytosis is basically a process that cleans up debris in the brain and damaged tissues and cellular debris and whatnot, and things that you just don't want in your brain. And the goal is to understand if nitrosylation of ApoE changes its specific function, microglia phagocytosis, in a harmful way and presents negative effects. Like, how can we alter this and fix this if this is the case? And in this picture, this is basically how phagocytosis works. In microglia, where this thing that looks like an octopus is the microglia, and chemoattraction is where chemotaxins, which are basically damaged tissues or unwanted debris, what will happen is that the microglia will move towards this chemotaxin, and then from there it'll engulf it or it will attach to it, where you can see it says tethering or engulfment, and then from there it'll break down that debris into different like proteins and cells so that it doesn't like float free in the brain. So that's how phagocytosis works. So at the beginning of our experiments, we used the model EAE, which is a mouse model, and a screen is done to look for proteins nitrosylated in this EAE model using mass spectrometry. And we noticed that a top hit from that mass spectrometry was ApoE. So this tells us that NO targets ApoE, which is very important because if it's targeting ApoE, we know that it has the ability to change its function of microglia phagocytosis. And if it does so in a negative way, that's not good. So we want to see if it does have those negative effects. So what we did was nitrosylation of these proteins, ApoE, was done. And to confirm ApoE is nitrosylated, biotin switch, which is basically a nitrosylation assay, is done in control mice and EAE mice, just to see if this really does have an impact. And we noticed that a band appears in the EAE model and not the control, which tells us that ApoE is in fact targeted by NO in EAE mice. So now we're trying to figure out, well, does it impact it negatively, or does it not impact it at all? And if it does impact it negatively, what can we do about this? So what we did next was an in vitro myelin protein phagocytosis uptake assay. So if you notice, the middle picture has a bunch of blue and green stuff, but basically these blue things are nuclei, and these green dots are debris that it's uptaking, because in phagocytosis, like I mentioned before, proteins like ApoE will conduct phagocytosis in that they break down cellular debris that's not wanted in the brain. So the more green it has, basically the more phagocytosis it's doing, and that's a good thing. But if you notice the picture on the right side, the ApoE SNO, this tells us that the ApoE is in fact nitrosylated, so NO has attached to the ApoE. But this means that, so because there's not as much green, that tells us that phagocytosis is in fact not happening as much, because there's not as much green, and that tells us not as much debris is being uptaken, which is not good, because that tells us that it's just not working, and it is in fact having a negative impact. And which is also shown in this graph, where there's a statistical significance here. And then the same thing here. So basically what I did over the summer was count a lot of these nuclei and for this specific assay. And the main graph that you really want to pay attention to is this graph right here, because 0 tells us that the green that you saw on the blue nuclei, that 0 tells us that we didn't see there's 0 green on those nuclei. So that tells us phagocytosis was not happening as much as we would have liked to. And that is because of the nitrosylation, which is like right here, ApoE SNO, the nitrosylation on ApoE. So that tells us that it is in fact impacting phagocytosis. So for the results, we're still in the process of collecting and quantifying data from these in vitro uptake assays. So we don't have definitive results, but we are, this is still an ongoing experiment, and we're still waiting to see the overall results to see if this really is playing a harmful role with the nitrosylation. And so what we can conclude though, and what we do know from our earlier experiments is that NO targets ApoE in the EAE mouse model, which we proved with the biotin switch assay, and it can negatively alter the function of ApoE, specifically its role in phagocytosis. So with that in mind, if with the future assays that we do, and this is in fact proven, what we can do is see if it is harmful, what approaches can we take to prevent nitrosylation so that the brain doesn't continue to get damaged, and that can be further researched. These are my references, and my research was supported by the NINDS grant and the Kornberg Lab, which is my PI, Dr. Kornberg, and Payam, which is the postdoc that I worked under. And I'd also like to thank the Neuroscience Scholars Program for giving me the opportunity to conduct research over this summer. And I'd like to thank Ms. Thomas for answering all my questions, and Dr. Brown, Dr. Asante, and all of the scholars who are here today. Thank you so much for your time. Thank you. And if you have any questions, please feel free to ask.
So what are you doing? What is the plan going forward to have more robust assays?
Okay, Dr. Brown asked that when you were displaying your results, you saw that one of your assays was successful. One out of the 3 assays was successful. What are your plans to change your methodology in the future to maybe have more success?
Yeah, I think just doing more experiments to make sure that this actually is statistically significant and not just at random is important. So just repeating these experiments is going to be important, but with the time over the summer, obviously that's not enough time. So that again will take time, and also perhaps looking at other ApoE because there are different types. So that also plays a role in how much phagocytosis happens. Because if you look at ApoE4, that in that model, phagocytosis happens the least, whereas in ApoE2 and 3, it happens the most in comparison. So that also, those factors also impact those results as well.
Thank you, Amy.
Yeah.
Any other questions? All right, I think you're done. Great job, Amy. Thank you.
Thank you so much.
Next up, we are going to have Beverly Tudela, who is here in person. And she is.
Hello, everyone. My name is Beverly Tudela, for those of you who don't know me. And this summer, I have had the privilege of working under Dr. Sherry Marvel at the Cognitive Neuropsychiatric Research Laboratory. And my research has been computing cerebellar structural volumes through computer software to associate risky behavior in HIV and drug-dependent users. So a bit of an introduction about the cerebellum. So a lot of times when we think about the cerebellum, we may think of its role in motor function. And so a lot of people think of gross motor skills. However, it doesn't only play a role in gross motor skills, but also in speech coordination and even the reward system. And so this brain structure, although small in size, is very important and unfortunately oftentimes overlooked by many researchers, especially within HIV and when looking at motor impairments or even reward. So the cerebellum has been found to have connections with the ventrotegmental area and substantia nigra, which play a role in reward processing, reinforcement learning, and motor control. And cerebellar neuronal activation as a result of operative behavioral tasks have been associated with reward-related activity in the dopaminergic circuitry. So essentially, the cerebellum is always taking in information and processing and self-correcting. And this is basically explaining that this is also happening when it comes to reward. So I have gathered a couple of figures from a study discussing the cerebellum and the reward system. So if you look at the y-axis, we can see value-driven attentional capture, and on the x-axis, premorbid non-planning impulsivity. And participants were given 2 tasks, and the first task incentivized participants to pick one of 2 flashing colors in exchange for monetary reward. And the correct color response to the stimuli earned the participant a higher value reward compared to picking the other option. When comparing value-driven attentional capture, there was a decrease seen 6 months later, interestingly, due to a learned attentional bias, the previously learned association between color and monetary reward. This is seen similarly in the second task when participants had to finger tap. And value-driven attentional capture was observed again, and results showed a decrease in attentional capture once again in the second visit due to a learned attentional bias, resulting in a slower reaction time and working memory. These figures once again show the cerebellum's role in the reward system and reward learning. And with this information on the cerebellum, and its connection with the reward system and impacts on behavior, I have been working this summer on computing cerebellar structural volumes with aims in the future of analyzing behavior, specifically impulsivity, and possible associations with cerebellar structural impacts with HIV positive patients. So I have been using computer software programs such as MRI-ProGL, MATLAB, SPM12, and SUIT in order to compute cerebellar structural volumes, and using MRI-ProGL and SUIT together to create cerebellar masks. And so we look at 4 groups: controls, drug-dependent users, HIV positive patients, and HIV drug-dependent users. And so this is a figure that shows everything represented in red is what will be computed as cerebellar gray and white matter volume and cerebrospinal fluid. And so in later steps, we're able to calculate the total amount of cerebellar volume and even each of the individual 34 lobules, those volumes as well. So this is a over-exaggerated, but a pretty clear representation of what a inaccurate SUIT cerebellar mask may look like. So we can see that the cerebellar mask is off, and when this happens, we may need to go back and hand trace each of the different slices within the cerebellum, which I've been very lucky not to really have to do this summer. But I've still learned how to do it, and SUIT is generally accurate, so I've been very lucky again. So general trends using SPSS, using a one-way ANOVA test, we didn't really find significant differences in cerebellar volumes across our 4 groups. However, using t-tests, we did find differences in cerebellar volumes between the HIV positive and HIV negative groups. So we found that there was greater cerebellar atrophy with our HIV positive group compared to the HIV negative group. And interestingly, we found that our expectations were greater cerebellar atrophy with our HIV drug-dependent group, because individually we have seen they both cause cerebellar atrophy compared to the HIV negative groups. However, our results showed greater volume compared to our HIV positive only group, but still greater cerebellar atrophy compared to the HIV group. And so our speculations point towards possible neuroinflammation caused by drug use. So it may seem like they have cerebellar volumes, greater cerebellar volumes. However, this is also similarly seen within the basal ganglia and other cohorts. So our limitations were that although our speculations point towards neuroinflammation caused by drug use, this cannot be confirmed yet due to our limitations with MRIs and limited resolution. So we can't really see what is being computed as cerebellar volume and what is being computed as neuroinflammation. However, that's not to say that those with HIV and drug dependence are not as impacted as only with HIV positive patients. And another limitation was our limited sample size, and going forward, we'd have to broaden this to accurately represent all of our 4 groups and have more accurate data. And with time limitations, I was not able to look into the behavioral aspects of how there may be associations between structural cerebellar changes in HIV positive patients, but that's something that I will hopefully be doing next summer. So I would like to give a big thanks to Heather Thomas, Dr. Amanda Brown, Asante, and my mentor, Sherry Marble, for all the support this summer. And here are my references. Are there any questions? Yes.
So you mentioned that your participants in the study were drug dependent. Do you think it makes a difference what drug they're on, or did you?
So I believe, and I could be wrong, they used cocaine, and I'm not exactly sure how this may impact anything. I would have to come back to you on that, but I do believe that was the drug most used with these patients. Mhm. Anyone else?
Well done.
Thank you.
Next up, we have Shaheen Amari.
I'm just going to share it. No, I'm just saying I'm going to share it. All right. Well, thank you all for being here today. My name is Shaheen Amari. This summer, I pleasure of working in the same lab that I've been in the last 2 summers, and it's the Wu lab. Okay. Can you guys hear me? Okay. Yeah. And the topic of my presentation is sleep and whether or not sleep is a unitary phenomenon in flies. So sleep is a natural, recurring, and reversible behavioral state where the body undergoes a lot of complex physiological changes, like changes in brain activity, cardiovascular system, immune function, and many other systems within the body.
Sleep is also a fundamental pillar of our overall health and vitality. Proper sleep is necessary for physical healing, emotional well-being, immune support, cardiovascular health, and on the flip side, inadequate sleep can lead to, it can undermine potentially all aspects of human health and performance. And virtually all animals studied thus far, ranging from simple organisms to complex vertebrate, engage in a sleep-like behavior. So this suggests to us as biologists that sleep has a fundamentally important role in survival and biological processes. Now, sleep is a dynamic process, and it has a cyclical nature with distinct stages marked by unique physiological activity patterns. In humans, sleep consists of two main states, which are the rapid eye movement, or REM, and non-REM sleep. And non-REM sleep itself is composed of three distinct stages. During a night of sleep, you cycle through all of these stages about every 90 minutes, and the non-REM sleep is more dominant early on, and as you progress through the night, REM sleep becomes more prominent. These are some typical polygraphic recordings of cortical activity, eye movement, and muscle tone during wakefulness, deep sleep, and REM sleep. And as you can see, each physiological measure exhibits distinct levels and patterns of activity, depending on the state of the body. Now, in fact, sleep stages themselves are determined based on stable changes in physiological processes, such as neural activity, body temperature, heart rate, and many other physiological processes. And though we have learned a great deal about sleep over the past several decades, there are still many aspects of sleep that we do not yet fully understand. And because it's such a complex process and it has a multifaceted nature, sleep remains an active area of research, a very exciting area of research to this date. And to study sleep, a lot of researchers use model organisms, and the most commonly used model organism in the field of sleep is Drosophila. And people often question, why would anyone study flies? And to that, I like to note that fruit fly research has led to some of the main scientific advances of the century in biological sciences. And these 5 individuals are scientists who received the Nobel Prize in Physiology and Medicine because of their work with Drosophila, which ranged in topics of the organization of the olfactory system, the innate immunity, and most recently, the molecular mechanism of circadian process, which is relevant to sleep because sleep is a circadian behavior. Now, the question is, how can you tell if an animal is asleep if it doesn't have eyelids? In the case of flies, we use a lot of different methods researchers use a lot of different methods to determine whether a fly is asleep. And the most commonly used method is monitoring their locomotor activity. And there are also other methods like assessing arousal threshold, looking at changes in metabolic rate, and measuring local field potentials across different regions of the brain. Conventionally, fly sleep has been thought of as a unitary phenomenon, and the field has been determining sleep as any period of immobility that lasts about 5 minutes or more. And this definition, just like it sounds like, it's not that precise, it's vague, and we don't know, not all 5 minutes of immobility can be confidently determined as sleep. So in the WU lab, we wanted to go beyond that, and we wanted to see if we can characterize sleep by looking at not just the behavior, but also the physiology of the fly. And by doing so, we wanted to determine whether fly sleep is a uniform process or does it also have stages like sleep in humans? Now, to that end, we started by looking at muscle activity. We monitored calcium activity in freely moving animals that were sleep deprived about 24 hours before. And we used a custom-built sideways microscope in order to then see the animals. And in this image right here, to the right, this white chamber is actually where we place the fly in order to then visualize it under the microscope. And we were actually able to see muscle dynamics associated with sleep. In this sample video that I'm about to play, any changes in brightness is indicative of calcium activity. So as you can see here, muscle activity in the distal tibia, which is the circled areas colored in red, is silenced for about 2 minutes during this quiescent period that the fly is in, which I think is pretty cool because this is very reminiscent of REM sleep in mammals, which we all go through when we sleep. And the reason I say that is because during REM sleep, we undergo muscle atonia or muscle paralysis. And this has never been identified in invertebrates. So we've been able to see this, and I think it's very fascinating. So we observed that flies exhibit bouts of muscle tone loss after a period of quiescence. And these bouts take place multiple times during fly sleep. So it also has that cyclical nature. And the average time from 58 bouts across 12 animals is slightly less than 2 minutes. The summer that I did this work, I monitored muscle activity in more than 30 flies, and they all exhibited these bouts. So this indicates to us that this is a robust physiological state that we've been able to detect. Now, having identified the state, we now want to find behavioral markers that are associated with the loss of muscle tone so that we can detect these bouts without having to look at calcium activity. And in order to truly establish this bout as a substage of sleep, we will need to do more in-depth experiments and analyses, like measuring the arousal threshold and metabolic rate to see whether this is a distinct physiological state. And ultimately, as the gold standard for any work in sleep research, you would want to look at brain activity and see if there are any changes in the nervous system that are linked to these quiescent bouts. Now, I would like to thank Dr. Mehmet Keles, who's a postdoc at the WU Lab, who mentored me and had me on board with this project. I would also like to thank the WU Lab. That's a picture of us. There are a lot of us. It's true. It's a true privilege to be able to work with such an incredible and supportive team of scientists. I'm very grateful for that. And lastly, I would like to thank the entire Johns Hopkins Neuroscience Scholars Program, Dr. Asante, Heather, Dr. Brown. You guys, with your mentorship over the last 3 years, have truly changed the trajectory of our lives. And I could have never dreamed of doing such cool research at such a fine institution. I'm very grateful for that. Yeah. So I don't know how we're doing on time, but I'm happy to answer your questions. Yeah, sure. So yeah, yes. So the question was, how do we sleep deprive flies? And so what we use is, it's a shaker that we first put the flies in a tube, and about 24 hours before doing the behavioral monitoring, we place the tube in a shaker that then you can set to the time intervals to less than 5 minutes to make sure that within every 5 minutes, the shaker shakes the animal and make sure that the animal is immobile. And that's how they actually sleep deprive the flies in a sleep state. Yeah. But it's a funny process. Brandon? Okay, so the question was how we visualize calcium in these animals. And so the way we did it in this experiment was we used a genetically encoded calcium, fluorescent calcium marker called GCAMP, which some of you might be familiar with. And we expressed GCAMP particularly in muscle tissue, which is why that's one of the benefits of doing genetic work with flies. And the reason is you can specify what kind of cells you want to look at. And that's how we monitored muscle activity with calcium.
Yeah.
All right. Thank you for listening.
All right, thank you everybody for your attention. That is our last talk before lunch. We will be back after our lunch and poster session at 2 p.m. To resume the afternoon, okay? Please also remember, if you haven't already, to view all of the posters if you're joining us virtually, and to make your votes for each of the poster assessments before 4 p.m. this afternoon, so we have time to figure out who's going to be winning our awards this afternoon. Thank you again all for your attention, and we will see you back here at 2 p.m.
Okay, we are back. Welcome back. We had a wonderful poster session. Outstanding job by everyone. Okay, so you see here all the information. Asante has been so good at reminding you about voting. Excellent. So yeah, we'll go ahead and get started. Did you want to? Go ahead. Thank you. Thank you so much.
No problem. Just to echo Dr. Brown, welcome back, everybody. Hopefully all of our virtual guests had an opportunity to see all of the great posters that everybody put up together. They all did a lot of really hard work on those. We're very proud of them. To start the afternoon portion of our session, we next have Ben Thompson. Come on up, Ben.
Thank you.
Right here. Yeah. Okay, there you go.
All right, good afternoon, everyone. My name is Ben Thompson, and today I'm going to be presenting my research project, Downstream Effects of CHIMP2B on the Nuclear Pore Injury Cascade of ALS/FTD. So ALS is a progressive neurodegenerative disease where your nerve cells gradually break down, reducing the functionality of your muscles. And so one part of this process is that the cell becomes dysfunctional in its nuclear pore complex and things can't get in and out of the nucleus. And so this injury to the nuclear pore complex happens to be an early indicator and contributor to both sporadic and familial ALS. But this injury happens in a sequence of events. And the first event is this localization of CHIMP7 in the nucleus. And following this localization, all the nuclear porins start to fall down in a sort of domino-like sequence, starting with POM121, and then the rest proceed. Once the nuclear pore complex is completely injured, there's this loss of nuclear TDP-43 function, and TDP-43 is important because it's a mass gene regulator. It regulates hundreds, if not thousands of genes, including STAPN2. So CHIMP2B is another, it's a different from the process I just explained, but it's still part of the same SCOR3 pathway that plays a role in many of the fundamental cellular processes, such as cytokinesis, exocytosis, et cetera. And it's expressed in all the major regions of the brain and every cell type of the central nervous system. And the reason that it's important is because previous research has demonstrated that a rare autosomal dominant mutation in this protein CHIMP2B has caused FTD. And so in more experimental work done in the COIN lab, which is the lab I've been working in over the summer, point mutations of single amino acid changes in CHIMP2B have been shown to show similar pathology in nuclear pore complex injury in ALS neurons, like I explained earlier. And so the end of this pathway is this TDP-43 dysfunction, which is what I'm going to take a further look at. So TDP-43 in previous research has been shown to that low levels of it lead to missplicing in many forms of genes. And this actually causes an increase in the expression of the truncated or non-functional form of many genes like STAPN2. And so previous research has also shown that this reduction in TDP-43 causes a reduction in the functional forms of genes as well. And as you can see on the bottom, low levels of TDP only lets binding to the first intron of STAPN2, creating a much shorter version of the gene. And so what is this what does this all mean? Well, lowering levels of functional TDP-43 increases this cryptic exon splicing and reduces the mRNA expression of normal genes. And also these point mutations in CHIMP2B that I mentioned present similar nuclear core complex injury to sporadic ALS and C9 ALS. So the sort of question I'm going to be asking this summer is like, what are the downstream effects of TDP43 in these CHIM2B mutants? And so this is sort of the experimental background. Single patient lineage, CRISPR-Cas9 is used to insert the CHIM2B point mutations, and these cells were obtained from the ND project. And so this is just sort of an overview of my experimental process. And the reason all the dates that we harvested genes had significance because of the pathological cascade that the cell goes through during ALS. So at day 18, that's when the CHIM7 accumulation happens in the nucleus. At day 25, POM121 is reduced. Day 32, MPC injury. And finally, day 46, which is the data I'm going to be looking at here, is the beginnings of TDP43 dysfunction, because that's when we see changes in gene expression. So this is just a list of the samples that I samples and target genes that I looked at this summer. Obviously, my control and the knockout, and then obviously you can see the different forms of the different mutations, what amino acid was switched at what position, or if there was a stop codon there, and then as well as the target genes. Some of them are functional forms of the gene, like ELA, VL3, and PFKP. And then there's also a couple cryptic exons that we tested, such as ACTL6B, that we would see that we would expect to see an increase of. So this is sort of the first data table. Pull up page pointer. So as you can see, the functional genes ELA, VL3, PFKP, ARCAN, and SELPLG, and SATHIN2 all showed a significant decrease in their expression. And reminder, these are all functional normal genes. So this is indicating that these CHIM2B mutations are actually showing downstream dysfunctional TDP43 effects. And on the other side, the truncated form of SATHIN2 actually showed significant increases in expression, signaling that these shortened forms of genes are actually being overexpressed in these mutants. And so beyond from that, two other genes, GFL2, which is responsible for like DNA repair, and ACTL6B, which is responsible for histone acetylation and chromatin condensation, also saw a increase in their cryptic exon version. In other words, there was a decrease in their normal form, which would be bad in a cell that needs to repair its DNA and to have the normal functioning nucleus and chromatin. So sort of the first, the conclusion of this part of the project was that the loss of functional TDP43 in human iPSNs results in an increase in the expression of these misspliced genes, and on the other side, a decrease in the expression of the normal functional genes. However, a wider panel of genes must be investigated, and we're still currently in the process of analyzing the qPCR results. So kind of before I go into the second half, I'm just going to talk about the difference between the iPSN model, the human neurons, and the mouse model. So in human C9 ALS, which is different from these CHIM2B mutants I'm talking about, a reduction in the expression of POM121 triggers a pathological cascade that decreases the expression of other nucleoporins. So same thing as regular ALS when POM121 falls first. However, the caveat is that in C9 ALS disease mouse neurons, this MPC injury actually doesn't happen, or at least doesn't happen in the same way. And so last summer I asked, can MPC injury in mouse neurons be induced? Essentially, if we knock down POM121, will the others kind of like fall as a result? And the answer was no. As you can see, the POM121 knockdown was successful, but nucleoporins 153 and 50 both stayed, there was an up expression of both of them in both the control and the C9 disease mice. So that sort of transitions to what I've been doing this summer with these CHIM2B mutant samples, because CHIM2B is, like I said, a member of this SCOR3 pathway and has been shown to have a role in nuclear biology. While it's not the same as C9 ALS, it has been shown to have very similar pathology to C9. Both C9 and CHIM2B have been shown to have like very similar pathology. So that's kind of how they're connected. And so what I did this year was a sort of examination of mouse brain sections of CHIM2B mutants. So we took two groups of 18-month-old CHIM2B mutants and stained them for both TDP43 and POM121, and another group of TDP43 and CHIM7. And so this is just sort of a overall process, just deparaffin, apply antibodies, image. And so this is sort of my, just like a select results of what I did. As you can see, the top two pictures are the diseased mouse neurons and their POM121 expression. And the bottom picture is a control. And then obviously the graph is showing the nuclear to cytoplasmic ratio of POM121. And as you can see, the diseased mouse neurons actually have more POM121 expression in the nucleus, which is surprising because we would expect, if these were human neurons, we would expect a much greater decrease in POM121 expression, especially at 18 months. So what this is again showing that this CHIM2B mutant ALS version is actually much different, or is actually the same as C9 ALS, but much different from human neurons. And so that's basically the conclusion, that these images suggest that the mutations don't have the same pathology in mice as they do in humans. And so future directions: continue analyzing qPCR, increasing number of duplicates to test for significance, and continue quantifying images of the brains. And yeah, so I just wanted to thank my mentor, Dr. Alyssa Coyne, for allowing me in her lab for 2 summers now. The rest of the Coyne lab, I want to thank Heather Thomas, Dr. Amanda Brown, and Dr. Ratnanathan. He's still in the Zoom here. Yeah, I'll take any questions. Yeah. How they what? Oh, yeah. Oh, sorry. Yeah. So she asked about, Dr. Brown asked about the specific point mutations of the CHIM2B protein mutants and how they could alter protein function. And so I just want to go back to this slide here. Some of the mutations, like this one, I29, they're all on different exons of the CHIM2B protein itself. And then you have this one here, this Q165X, X being like, there's a stop codon there. So all of these point mutations have been shown in previous literature. They have been shown in ALS lineages to have mutations at these points. So that's how they were first picked out. And then obviously once they realized that, they could detect like what specific amino acids were changed. So like I said, Q165, that's like a stop codon is put in place at the position 165. And I believe it cuts off like 49 amino acids and basically makes the protein not functional. Oh, I'm not sure about that. I'll have to get back to you on that. Okay.
Okay, next up, we're gonna have Jada Comendria. Jada, come on up.
Hello, everyone. My name is Jada Pamengia. I'm a JHNeurophyte Scholar in the Brain Health Program. And today I'll be presenting on effects of childhood trauma on facial emotion processing and people living with HIV. You look at this person's face, and what do you see? You take in the drooping eyes, the pinched eyebrows, and the slight pulling down of his lip corners. You'd say this person is sad. Our brains decode facial expressions by adding up sets of key muscle movements in the face of the person we're looking at, almost instantly. While you may not have realized that's what your brain was doing, I'm sure everyone in the audience recognized this person as sad. Now, the brain is a major target for HIV, with the infection wreaking havoc on the frontal striatal regions of the brain, which is seen here, despite antiretroviral therapies. This can cause cognitive and psychological dysfunction, including disruptive facial emotion disabilities. The parts of your brain that were so quick to place the facial emotion as sadness in that last slide may be damaged in people with HIV, and now a sad face could be happy or convoluted. But what happens when other comorbidities stack up? People with HIV have been found to have a greater burden of mental health issues, including substance disorders, depression, and PTSD. In particular, people with HIV are more likely to have a history of trauma. Recent studies have shown that the prevalence of trauma is 1.5 to 2 times greater people living with HIV in the general population. More than that, HIV infection and trauma occur together to produce a combined greater cognitive dysfunction associated with HIV. This brings us to the goal of my research, to address how childhood trauma in people with HIV is associated with poor cognitive function, particularly with facial emotion processing, a social cognitive task necessary to communicate, interact, and even to survive. I hypothesized that HIV-positive individuals that have greater burden of childhood trauma, specifically emotional abuse, should show worse performance perceiving facial emotions when compared to a control group. To conduct this research, we examined 151 participants that were recruited as a part of a larger study in the Brain Health Program titled IMPACT. This study is delving into the neural mechanism of HIV-associated CNS dysfunction despite viral suppression. These participants were enrolled and recruited from patient populations in the Baltimore, Washington metropolitan region from late 2019 to mid-2022. Subjects underwent a series of examinations, standardized demographic assessments, behavioral and medical screening, and cognitive testing, including the facial emotion perception task. For this research, we analyzed data from a single time point for each participant in the ongoing study. The way the facial emotion perception task, or FEPT, works is that the participants are presented with computer morphed images derived from the facial features of real individuals, each showing specific emotion. Each face is displayed on the screen one at a time, and then immediately covered up to prevent residual processing of the image. The participant must select which emotion the face is displayed from 5 options: fearful, angry, happy, sad, or neutral. The speed by which the participants gave their answer, as well as the accuracy, was documented. Participants also completed the Childhood Trauma Questionnaire, or CTQ, to assess burden of childhood trauma among our participants. Through the CTQ, we were able to assess trauma within 5 subcategories: emotional neglect, emotional abuse, physical neglect, physical abuse, and sexual abuse. Looking at our patient demographics, we had 56 controls and 95 individuals with HIV. In both groups, the average patient age was in the late 50s. Both also had a great percentage of Black and female populations, although not by much for the latter. And in both groups, most participants have been educated past high school. We had no significant differences between the groups, indicating that the demographics were controlled for. In terms of our data for the facial emotion perception task, we first want to confirm that facial emotion recognition is impaired in people with HIV. Looked at accuracy by facial expression, meaning what percentage of fearful faces were said to be fearful, angry faces said to be angry, etc. Looking at this graph, with the controls being in blue and the HIV positive being in red, we found that for fearful faces, angry faces, and sad faces, there was a significant difference in accuracy across groups. The controls performed significantly better in each instance. We also took a look at reaction time for both correct trials and incorrect trials, meaning how long it took for participants to choose the correct answer, how long it took the correct participants to choose an incorrect answer, respectively. We found that with happy and sad faces, HIV individuals, it's typically slower to give typically more time to assign them as happy and sad when given the correct answer. We also found HIV positive individuals were typically slower to give an incorrect response when faced with fearful and happy faces. All in all, this data does support facial emotion recognition impairment in people with HIV. To examine how childhood trauma will worsen performance on this task, we looked at how the presence or absence of the different CTQ abuse subcategories sexual abuse, physical neglect, emotional abuse, and physical abuse influence performance on the FEPT outcome variables, which again were accuracy and reaction time for correct responses, all while controlling for age, sex, race, ethnicity, and education level. This was done for both the HIV positive group and HIV negative group. Looking at both the models for accuracy and reaction time for correct responses, we did find there to be a significant effect of participant race in the HIV positive group on accuracy, shown on the left. And we also found a significant effect of race on the HIV negative group for response time, shown on the right. As we see in the least squares graph for both outputs, there is an outlier that is dragging the overall group performance. In the case on the left is this red group, which is the American Indians. And in the case on the right, it is the Asians, and that is an outlier. And these outliers will make up a handful of our participants. So while this does weaken the effect of race on FEPT performance, it does point out a flaw in our task. The face database for the FEPT is all Caucasian, and research does show that there are differences in speed and accuracy when it comes to identifying emotions in a race other than one's own. This would be why we see inconsistencies across groups. Additionally, for the HIV negative group, the presence of emotional abuse did have a significant effect on facial emotion perception tasks in terms of response time. However, we did not find any significant effects of abuse exposure as measured by the CTQ on FEPT test performance measures for the HIV positive group. What does this mean? Well, the reason we may not be seeing any effects for HIV positive group could be due to some limitations of this study. There's a small sample size, which could skew the results. Uniform face of the task-based database, as I mentioned prior, but also cause differences in task performance, and a relatively low incidence of trauma in the CIS sample compared to others may mean that our data is not as generalizable. Childhood trauma is typically prominent in people with HIV, but in this study, we found low average scores on the CTQ for both groups. Some other limitations of this study is that due to the nature of the facial emotion processing task, may not be representative of specific emotions. The task used to measure facial emotion processing uses static faces, which are contrary to real life faces, which are dynamic and have a range of expressions to demonstrate an emotion. Another limitation that the control group may not be a true control group, as people with HIV also experience comorbidities such as depression and anxiety that may also influence performance on the facial emotion perception task. Additionally, while the majority of our participants are Black, the faces in our facial emotion processing task are white, which may have affected their results due to again, the cross-racial identification differences I mentioned prior. Fortunately, this research is a subset of an ongoing longitudinal study. So as additional data is collected, we may find more interesting results. We obtain imaging data from each of the participants. This may be very revealing as brain morphology is also affected by childhood trauma. These results will go to inform clinical settings to create more personalized treatment plans for individuals with HIV who have experienced childhood trauma. I'd like to thank NIMH for funding this study, Olivia Fox and Hannah Lee for data access, Dr. Asante, Dr. Brown, and Ms. Hedder for making this research experience possible. Thank you for your time. So the question was, when they make mistakes while completing the FEPT task, is that due to voter mistakes or, sorry? Sorry, to clarify. Okay, but that's a really interesting question. I didn't think about if that maybe motor deficits. I was more thinking that maybe the mistakes happen maybe because of cognitive deficits. That's really interesting take, and definitely would like to delve into that. Not quite sure. Your typing task and like. Oh, okay. Digit test. Okay. Mhm. Okay, yeah, we do have that task as part of our content database. All right, great job, Jada. Thank you. Next up, Bethel Jebaselassie. Come on up, Bethel. Okay.
Hello, my name is Bethel Gebrselassie, and I will be talking about interdisciplinary care with patients with complex comorbidities. So I had the honor at working at the Johns Hopkins Hospital at Myers 6, which is an inpatient unit at the Johns Hopkins Hospital, treating hospitalized patients. And I worked at the Bartlett Specialty Clinic, which treated with outpatient facility within hospital, providing specialized care for people with AIDS and HIV. I also had the privilege to work under Dr. Glenn Trisman, who is an esteemed psychiatrist who has led the unit for more than 35 years. And one of my favorite quotes is, "He who saves one life is as if he saves the entire world." So for the methodology, we got to conduct rounds every morning in July. And we conducted observations and participated in rounds, which are similar to patient care interviews. And during these rounds, we closely observed doctors and nurses interacting with patients, assessing their condition, and making treatment decisions, actively listening to patients' symptoms and seeing their progress during rounds with valuable information. And the aim of this discussion is to analyze the patient's prolonged illness and examine the influence of his over his life. So we're going to be talking about patient Z, who is a patient I was able to see come in during the rounds and also leave, which I think is a blessing, given that there are people there more than weeks, several months. And our hypothesis was this autonomia-induced regulation of the patient's autonomic nervous system, and that led to constipation due to heightened sensitivity, which we call allodynia. When we're talking about patients with challenging disorders and uncertain outcomes, let me just go back. When we're talking about patients with intricate illnesses, we also look at 3 factors, being that 1, we have technological advances nowadays to help these patients, being that breast cancer is treatable and people being able to lengthen their lifespan, but also if we have a patient who comes in very sick, for example, let's say HIV, we don't want to invest in them and help them only 75%, whereas we could commit resources and money and time to fully get them well, 100%. Let's say we help them only 75% and they were able to go out and infect 8 other people with HIV. Now we're dealing with 8 patients, whereas we could have initially rooted out the problem initially by funding it properly and making sure they're fully well. And lastly, the fundamental ethical obligation of the medical profession to help vulnerable patients who are sick and to commit to fully getting them better. So here we have patient Z. He is a 39-year-old male who is highly functional with a successful career, marriage, and kids. He has a recurring illness with dysregulated autonomic nervous system causing constipation allodynia, which is pain sensitivity. And some of the things we diagnosed him with was neuropathic and chronic abdominal pain, postural POTS, bipolar 2, gallbladder hyperkinesia, which is epigastric pain, psychiatric disorder, and cannabis and opiate use disorder, meaning he was also taking marijuana to kind of deal with the pain. And he was disabled because over the span, he would get pain and he would come in. Mind you, he has allodynia, so every time he eats, the body senses the overstretching of the stomach is pain. So now that he's feeling pain, he goes to the streets and gets fentanyl. Now you might ask why a man who is very successful, who has kids, is obtaining fentanyl from off the streets. And that is because he is an unstable extrovert, which we will get in the next slide. But this causes him to take high-risk decisions, resorting to obtaining illegal fentanyl use despite responsibilities and having a family. Emotional influence, decision-making heavily influenced on emotions, which we will talk about. Trait vulnerabilities, being that he exhibits high risk of openness and predisposition to continue medications. So he comes in the hospital very sick. Mind you, his stomach is hurting, and we stick with him. We treat him, we get him better, but the moment he steps out, he thinks he's getting better from the pill, so he stops, therefore continuing a reoccurring cycle of coming to the hospital. And there has been 3 times where he came in for this over either 6 months or like he's just been in pain for a very long time. And the current presentation is he had subacute worsening of chronic pain due to self-discontinuation of medication. When we talk about a patient, we would describe him as an unstable extrovert. This is the 4-quadrant personality model, also used by the Greeks, and we would describe him as caloric. He's an unstable extrovert. So you would look at quadrant 1. He was restless, aggressive, excitable, impulsive, optimistic, and active. I chose this patient because he fit an unstable extrovert perfectly. And given that my time there during rounds, I saw all of these characteristics, being that he was very aggressive. If he thought a nurse looked at him a certain way he didn't like, he was very angry and riled up, and he would, like wait until I see, like he was just very angry. But he was also impulsive, but also optimistic. And this is where the reoccurring cycle comes in. He came 3 times, as I said, because he gets into self-medicating. He feels pain. And a normal person, you feel tired, you feel pain, you would go home and take Tylenol. But he went to the streets to get fentanyl, being that he's impulsive and optimistic. So every time he comes to the hospital, he's very optimistic, thinking I'm going to get better. And, once I leave, I'm not going to continue this, but then there's a continuous cycle. So the results that we got from this patient is he had dysautonomia, he had major depression and unstable extroversion. And characterizing, this is a challenging temperament just because given that we were able to know what his type was, we're able to diagnose him better and know how he's going to kind of deal with the treatments and like how he's going to react. Okay, and majority of individuals have a balanced mix of temperament with a mix of introversion and extroverted traits, allowing effective adaptations to various situations. Extreme extroverts may struggle with quick decisions, while extreme extroverts may act impulsively without considering consequences. So our guy, patient Z, felt pain, so he decided, to deal with it in a very extreme way. And understanding these temperamental styles improves communication without considering consequences. And incorporating Incorporating patients' temperament into healthcare can lead into more successful outcomes and better patient-provider interactions. And here's a graph of, you see two bell curves for instability, stable, and then extroversion, introversion, and we see there are two extremes. We call the people on, for example, the right hand of the x-axis, we would call that like 2 scoops of feeling. So people who feel emotions very easily. Conclusion. So as we see, the medical system faces a significant problem in providing adequate care for patients with complex patients. Prolonged illness causes disability and strain on families due to the lack of coherent treatment. Atypical patients requires doctors with broad knowledge, hence interdisciplinary care, and willingness to think outside of the box, incorporating interdisciplinary approaches from various disciplines. This approach leads to better care and improved outcomes for complex patients, and also incorporating, when we talk about getting a holistic view of the patient from initially diagnosing them, we're talking about medical, psychiatric, and psychological factors, seeing how they interact with other people in their lives and certain temperaments. Yeah, I would like to thank Dr. Cheeseman, who was my doctor. He was amazing at teaching, guiding, and making me feel really part of the team, even though I'm an undergraduate. And I'd like to thank Dr. Brown, Dr. Mokwala, and Heather Brown. Thank you so much. Any questions? Okay, so dysautonomia, we're talking about GI dysmotility. So the way his intestines and the way he takes food, and he's feeling epigastric pain. So if he's eating food, the way he reacts to it is not as a normal person would. Did that answer your question?
Second question, actually, what was like, so as you observe the patient, what was the patient and how the patient speak over time with like the reaffirmed meeting that you have with the patient? So you mentioned the patient came back to the to you guys several times. So how did that change in the process?
Okay, so the question was, what did I observe about the patient and how did it change over time? When he initially came in, I think he had prior experience with my doctor, which I didn't know him, but from the way they interacted, it was years ago. And I think he came in for a similar thing. He's had pain that he's went to doctors, like he's had pain for about like 6 years. And so he was traveling from doctor to doctor trying to figure out what was wrong with him. But then when he came in, we were able to kind of find out what was wrong with him. And then from there, see how he's kind of dealing with the medications that we gave him. And then how I kind of saw his temperament change. I think he was always aware of what was going on, but as he was leaving, I noticed he was very optimistic. And as I said from the chart in quadrant 1, optimism is one of the character traits where for unstable extroverts, where you feel like, hey, this time is going to be different. And you get optimistic, but the moment you step out, you feel better and you're like, I don't need these pills anymore. And then it's like a continuous cycle. So kind of understanding where he kind of lies to help that issue. Hope that answered your question. Any more? Yes. Yes. It's different. I think bipolar 2 is more, it's more extreme in regards of emotions and how they tend to react to people.
him if he was given any type of medicament for opioid use disorder, or if it was just straight up withdrawal from.
So he wasn't feeling withdrawal when he came in because he was still using, but he told us he was feeling pain. So what we do for those cases is we usually taper them off. So we go from the full amount, and then maybe in 2 days, we're gonna half it.